Welcome everyone. In today's lecture, we are going to cover plant growth and development in one shot. It comes in Plant Physiology, 11th NCERT textbook. It is chapter number 15, Plant Growth and Development. Let's start the chapter, right? So it will be very helpful for you because it is one shot lecture and each and individual point of NCERT will be covered here. It will be useful for your NEET examination also as well as boards also. Now when we start with plant growth and development, in the first paragraph itself, the author has asked us some questions. Let us look into those questions and then let us discuss what is growth and what is development. So here are the questions which they have given in NCERT. I already took it on the board. Now the first question, from where does the root, stem, leaves, flowers, fruits and seeds arise? From where will all the parts of the plants will arise, they asked. So we know it's embryo, right? From embryo only all the plant parts will originate. So what is the answer here? So the answer here will be embryo, right? And that too in an orderly sequence, in a defined manner, means that is called differentiation, right? Second question, are you aware of the terms seed, seedling, plantlet and a mature plant? Yes, we know what is seed. Seed is the end product of sexual reproduction. Fruit and seed are the end products of sexual reproduction. Now then seed, if we keep the seed inside the soil, here is the seed which has kept inside the soil, it started germinating. The germinating seed is called a seedling. The germinating seed, see here the root development has started. So this is called seedling. Now the seedling further grows into a tiny plant which is called as plantlet. Now here this we can take it as a plantlet. So seed grows into seedling, seedling grows into plantlet. Now that tiny plant grows massively. It develops its root, shoot, leaves, flower, fruits and all those things which is called as a matured plant. So we are aware of the seed also, seedling also, plantlet also, matured plant also. Third question, trees continue to increase in height or girth? Yes, so plant growth is different from animal growth. Plant growth is unique, we will tell. It is indeterminate, we will tell. It keeps on growing from birth to death. Such a type of growth is called indeterminate growth. So trees continue to increase in height because of the presence of meristematic tissue. They keep on growing. That growth which increases the height of the plant is called as primary growth. And sometimes growth happens only in the trees, dicot trees and gymnosperms. In the later stages, the plant grows thicker. It increases the girth of the plant, diameter of the plant. Then that is called secondary growth. So trees continue to increase the height and girth. So increase in height, what is it called? It is called primary growth. Whereas increase in girth, what is it called? This is called secondary growth. Right, next question. But leaves, flower, fruits of the same tree have limited growth. Yes, so only root and stem parts of the plant will show indefinite growth. Whereas if you see the leaves, after growing to a particular dimension, they will stop. Fruit also, reaching a particular size, it will stop. So leaf, fruit and seed, which are also the parts of the plant, they have limited growth. Yes, the leaves, flowers and fruits of the same tree have limited growth. Means like us, it is called definite growth or it is called determinate growth. It is called determinate growth. Next question, why does vegetative phase precedes flowering in the plant? Why does vegetative phase precedes flowering in the plant? Now what are the vegetative organs? Root, stem, leaf are the vegetative organs. So why the plant will grow first? It will develop its root system, shoot system and leaves. Then only after reaching a particular growth stage, it starts flowering. Why will the vegetative phase precedes flowering? Means for flowering it has to accumulate so much amount of energy. So only when the leaves are developed, only when the leaf will do photosynthesis, it can get that food and energy, right? So that is why first it will ensure the nutrition, then it starts flowering. What is flower? It's a reproductive part of a plant. That flower, if it has to flower, it has to produce pollen grains and egg. It needs so much amount of energy. So in vegetative phase it will generate that energy. So to ensure nutrition, we can tell to ensure nutrition. Next question. Is there any relation between the structure of the cell and the function it performs? Here I'll take the example of an animal cell. Animal cell, if you see neuron, so neuron has a long axon. Why is a neuron long? Means because nerves help in conduction, right? Nerve impulse conduction from one part of the body to other part of the body. So if it is long, if it is having branches and all, it can easily transmit the nerve impulses. That means the structure of a cell is related to its function. If you see plant epidermal cells, they are barrel shaped, they are rectangular. So why they are rectangular? So one rectangle and another rectangle can be perfectly fitted in this manner, so that there will not be any gaps and all. If two circles are there, between two circles certainly gap will come. So that means the shape and structure of a cell are related to its function, right? So is there any relation? Yes, there is relation, right? Next question. Can the structure and function be altered? Yeah, we can alter the structure of a cell and the function of a cell. Now we say that differentiated tissues which are doing their function, now during the process of dedifferentiation they become meristematic again. They become meristematic again, they divide again, they make a new tissues and they'll take up another function. So that means, can the structure and function of the cells be altered? Yes, you can alter them. Means a mature cell can become meristematic again. A mature cell which has lost the ability to divide can become a dividing cell, meristematic again. So here we can give the answer, yes, we can do. Now next question. All cells are descendants of zygote only. Now male gamete fuses with the female gamete, then that's called syngamy. Syngamy leads to fertilization. Fertilization leads to development of zygote. All the cells, this zygote only will cleave and it will make entire organism. So all the cells are descendants of zygote only. But why? They have different structures and functions. Why some cells are different, some cells are different means depends upon the location. If it is a peripheral cell, it has to do the protective role. It takes that particular shape. If it is an innermost cell which is a part of xylem and phloem, then it has to do conduction. So depending upon their locations, they differentiate and they'll take up the function. Even though all the cells comes from a single cell stage called zygote, they undergo differentiation. They move and to their places and then they take up the function, right? So we solve all the questions which are there in the first para of the chapter. Now next they tell that development is sum total of two processes, right? So they're telling development is sum total of two processes. One is growth, other one is differentiation. First a plant grows. Means it increases the number of cells, right? It increases the number of cells. Then it differentiate into root, stem, leaf, flower, fruit. That means development is sum of two processes. One is growth. After growing, they will differentiate, right? Now next, they have given in NCERT, in the next page, they have given this picture showing like how a seed develops into a plant. How does a seed develops into a plant? So this is a soil surface. Under the soil surface, they sowed a seed. They kept a seed. Now this seed started germinating. I told you the germinating seed is called as seedling. Now this is seedling here. Now in this concept, we can talk about two types of germination. One, as I wrote here, it is apigeal germination, and the second one is hypogeal germination. Let us see the differences between apigeal and hypogeal with examples. In apigeal germination, as we can make out here, shoot system did not start it, but the root system is developing fast. Okay, so this is an embryonic axis which came out, which will develop into radicle, and the root system is called hypocotyl, right? Hypocotyl will make a radicle. This is called hypocotyl. The hypocotyl growth is more here. Since the hypocotyl growth is more, it pushes the cotyledons, it pushes the seed above the surface. So that cotyledons came above the surface. It is because of overgrowth or the fast growth of the root hypocotyl. So in this picture we can see the cotyledons came out and the seed coat has opened. the cotyledons have separated. Now in between those two cotyledons, epicotyl is coming out now. Epicotyl is coming out. It made a hook-like structure which is called epicotyl hook. Like a hook it comes and then it opens out. So this particular portion is called epicotyl. These two are the cotyledons of the seed and this is a seed coat. In the third picture we can see the seed coat got removed off, the cotyledons are well separated and the plant has made its first leaves. The plant has made its first leaves. Now when the first leaves are made, then the plant can do photosynthesis. Till then it was depending upon the nutrition from these cotyledons. Cotyledons have stored food. From these cotyledons the plant is taking the food. When first leaves will come, they will do photosynthesis. Then there's no role of cotyledons. So that's why in the fourth picture we can see there are no cotyledons. Cotyledons fall off. Okay. So this type of germination where hypocotyl grows faster, it pushes the cotyledons out, above the soil surface if the cotyledons are seen, then that is called epigeal germination. Example for epigeal germination are, one is bean we can take, this example is given in NCERT. Second example, I just wrote few more examples. Castor is also the example, all the gourds, bitter gourd, ridge gourd, right, all those things. Castor, gourds, cotton, bean are the examples of epigeal germination. The other type of germination where cotyledons will be remained inside the soil only, so that is called hypogeal. Hypo means inside, epi means above. So in hypogeal germination, this is the seed. In this seed if you see, now the root axis is this much only, but the shoot axis is this much. That means what do we say? This is hypocotyl, this is epicotyl. The epicotyl is growing more here. The epicotyl is growing more here. Since the epicotyl grows more, then the seeds will be inside only the shoot comes above. So the cotyledons are remaining inside only and the plant will develop. That germination is called hypogeal germination. Example is P. Once they ask this in AIMS examination, give the example for hypogeal germination. The answer is P. Example for epigeal germination is given in NCERT. They have given bean as an example. The other examples are, we can take castor, we can take gourds and we can take cotton also. So take a screenshot, then we will continue. What is growth? What is the definition of growth? Right. Now let us see. The chapter is about growth, right? Now let us see what is growth, what are the characteristics of growth, and what is the definition of growth. Now if you want to interact, any comments, yeah, you can post them in the comment box. Any other help you need related to NEET examination, yeah, you are welcome. You can text them in the comment box. Now let us start. What is growth, right? Now come on, tell me, growth is most fundamental character, right? So living organism means it keeps growing. So growth is regarded as one of the most fundamental and conspicuous character, means visible character. You planted a small plant, after 1 week it will grow this much, means it is visible for you, it's clearly visible for you, this much it has grown. So growth is one of the most fundamental characteristic and visible characteristics, conspicuous characteristic of living organisms. Now if you have to give the definition of growth, growth can be defined as irreversible. What do you mean by irreversible? Permanent. What do you mean by permanent? Now we are grown up, we are adults, we cannot go back to our childhood. That means it's irreversible. We can't go back, right? So that means it is irreversible, it is permanent. So what happened? When we are born, we are this much. Now we have grown this much, right? Means there is increase in height or weight or the size of the organ. So the definition of growth goes like, it is defined as irreversible, permanent, right? It is defined as irreversible, permanent increase in the size of the organ. Increase in the size of the organ. So children, this is a definition of growth. Now growth, if it has to happen, it should be accompanied by metabolism. What is metabolism? Metabolism is the sum total of catabolism and anabolism. Catabolism means breaking the food. When you break the food, you get energy. Using this energy, you are building your body. Plant is building its body. That means growth is accompanied by metabolism. Metabolism means which includes catabolism and anabolism. So the next characteristic is growth is accompanied by metabolism. Now what is metabolism? Metabolism is a sum total of catabolism and anabolism. Now what is catabolism? Catabolism is breakdown. And what is anabolism? Anabolism is synthetic process. Catabolism is breakdown and anabolism is synthetic process. Now when you break any compound, we get energy. Using that energy, the plant will do anabolism. It will synthesize. It will make its body. Okay. So that means growth occurs with energy. If energy is provided, then only growth will happen. That means growth occurs at expense of energy. Right? Growth occurs at expense of energy. When you provide energy, then only growth will happen. So if we have to give the examples of growth, now expansion of the leaf, expansion of the root, expansion of the stem is an example. Now if you have to see the example, in NCERT they have given expansion of the leaf surface is an example. Expansion of leaf surface is an example for growth. Now in NCERT, the author has asked at this point like swelling of the wood, what do you consider it as? So when you drop a wood block in the water, it swells and it increases its weight. So then what is it called as? It is growth or it is imbibing water? Swelling of wood is not growth, children. Swelling, wood is a non-living substance. Non-living substances will not grow, only living organisms will grow. Means swelling of the wood is not an example for growth, it is example for imbibition. So swelling of wood, what is it? This is called imbibition. It is not growth. Okay. Now we can say that plant growth is different from animal growth. Plant growth is unique because the plant have meristematic cells. These meristematic cells will allow the plant to grow from birth to that. Continuous growth will happen. Such growth is called indeterminate growth, indefinite growth. So that means we can tell plant growth is unique. Why is it unique? Because of the presence of meristematic tissue. Due to the presence of meristematic tissue. Right? Due to the presence of meristematic tissue, it keeps on growing. So if it keeps on growing, plant grows continuously. Right? It grows continuously. Such a type of continuous growth is called indeterminate growth. What is this called? It's called indeterminate growth, or it is called indefinite growth. Whereas our growth till certain age, till 16 years, 18 years we grow and we stop. So that means we have a definite growth, whereas plants exhibit indefinite growth, indeterminate growth because of the presence of meristematic tissue. What are meristematic cells? Meristematic cells are self-perpetuating cells, means self-replicating cells, actively dividing cells. What are meristematic cells? They are actively dividing cells. In NCERT they have given they self-perpetuate, they'll self-multiply. In NCERT they have given they will self-multiply, they keep on growing, they actively divide, so that's why they increase the growth of the plant. Right? Now in this manner, when the new cells keep on adding, such a type of growth is called open growth. We can tell that the plant growth is open type. What do you mean by open type of growth? Open type of growth means where the new cells keep on adding. So in this they make new cells continuously. If they are making new cells continuously, then we have to call it as an open growth, right? Open growth, why it is possible? How it is possible? It is possible because of the presence of meristematic tissue. Now, meristematic tissue, suppose think, because meristematic tissue is there, it's keeping on growing. What happens if the meristematic tissue ceases? If the meristematic tissue stops to divide, what will happen? Plant growth will not complete and plant might die. In NCERT, they have asked a question here, what happens if the meristem ceases to divide? What happens when meristem ceases to divide? This was the question they asked in NCERT. Now the actively dividing tissue, if it stops to divide, then the plant growth becomes incomplete, leads to the death of the plant. What happens? The answer is plant growth is incomplete, then it is incomplete and then the plant dies. Right, the plant dies. So they're asking, does it ever happen? The next question what they've asked in NCERT is, does it ever happen? Right, it happens. It happens. You soak so many seeds inside the soil, all the seeds will not germinate. They cannot establish themselves because there the meristematic cells stop dividing. So does it ever happen? Yeah, it happens when the seeds fail to germinate. When seeds fail to germinate, we can see this. Now what did we see so far? We were discussing what is growth, the definition of the growth and the examples of growth, and wood dropping inside the water. If it swells, it is not growth, it is imbibition, taking up the water. And how is plant growth different from animal growth, that also we have seen. And why it is showing continuous growth? Because of the presence of meristematic tissue. Now the meristematic tissue, it is present at the root tip and the shoot tip. If the meristematic tissue is present at the root apex and the shoot apex, then that meristem is called primary meristem. That growth is responsible for primary growth. Primary growth is responsible for increase in the length of the plant. So the meristem which is present at the tip of the root and the tip of the shoot allows the plant to grow like this, increasing in the length. That growth, what is it called? It is called primary growth. Whereas in dicots, only in dicots and in gymnosperms, dicots and gymnosperms are trees. Now what happens later in their life? They start increasing their diameter also, they start increasing their width also, girth also, by developing another tissue, by developing lateral meristem, by developing vascular cambium, cork cambium, which are responsible for increasing the girth of the plant, girth of the tree. What is that called? Secondary growth. Take a screenshot, we will continue. Right, so we are telling the root apical meristem and shoot apical meristem are responsible for primary growth, which increases the length, of axis. Whereas in gymnosperms and in dicots, there will be increase in the girth. Right, so what is that called? That is called secondary growth. Root or shoot apical meristem, root or shoot apical meristem, what are they responsible for? Which type of growth they are responsible for? Primary growth, which increases the length. So they are responsible for primary growth. What do you mean by primary growth? Which increases the length of the plant. Which increases the length of the plant. Whereas if we take dicots and gymnosperms, these are trees. If we take dicots and gymnosperms, later in their life, they develop another meristem. Now, what is that meristem is called? It's called lateral meristem, secondary meristem. Later in their life, they develop secondary meristem. They develop secondary meristem. This secondary meristem is responsible for increasing the girth of the plant. It increases the width or it increases the girth of the tree. Now what is this type of growth called as? It is called as secondary growth. Example for secondary growth are development of vascular cambium, development of cork cambium. Examples of secondary growth are, so what are they? Developments of vascular cambium, we have seen it in anatomy chapter children, and development of cork cambium. So these are examples where they develop from lateral meristem. Understood? Now next, the author has asked, growth, is it measurable? Yeah, I told you, you dropped a small seed and this much plant came. After 1 month the plant became this much. You can measure it. So that means growth is measurable. Growth is visible also, growth is measurable also. Now what is that instrument which we use to measure the growth? What is that instrument which we use to measure the growth? There is a microscope which is called as horizontal microscope. The first technique is horizontal microscope. It will be fit with a scale and there will be a point. We'll keep that point. After 1 week we'll see from here to which level it has increased, we will measure it. That is called horizontal microscope. There's another instrument which is called as Pfeffer's oxynometer. That instrument was developed by a scientist called Pfeffer, so we call it as Pfeffer's oxynometer. Pfeffer's oxynometer is another instrument which measures the growth of the plant. There's one more technique in NCERT. They have given us a seedling and the growth of the root. That is called parallel line technique. What is another technique? Parallel line technique is another technique. In parallel line technique, in NCERT, they have shown us the picture in this manner. Like this they have shown, right? And A, B, C, D, E, F, G. Till how much they have given? Till G they have given, right? So they're telling this is A, B, C, D, E, F, G. Now earlier the seed is this much, later the seed became like this. Now if you see, the tip cells means it was this much. The distance between A to B is very less here, but the distance between A to B has increased here. The distance between B to C is again less here, but here the distance between B to C is increased. C to D is also increased. Means the tips are elongating more when compared to, see, the distance between E, F and G is still less only. So that means the growth is growing in this manner. So what is this line called? This technique of measuring the growth is called parallel line technique. In this parallel line technique, they told A, B, C, D, they have grown more when compared to E, F, G. When compared to EFG. This is one instrument to measure the growth. The other instrument is called Pfeffer's oxynometer and the other instrument is called horizontal microscope. So growth is measurable. Take a screenshot. We will continue. Now, in plant, at cellular level, so if you have to see the height of the plant, yeah, you can tell. But if you have to see the cell is increasing in its dimension, then how can you measure it? Means we have to measure the protoplasm. At cellular level, growth is increase in protoplasm. At cell level, it's called cellular level. At cellular level, so growth is, what is growth? It is increase in protoplasm. It is increase in protoplasm. So how can you measure the thing which is there inside? So increase in protoplasm is difficult to measure. Increase in protoplasm is difficult to measure. So that's why directly we can't measure. So if you want to measure, you have to tear the wall, you have to tear the membrane, and it will die. So it is difficult to measure directly the increase in protoplasm. So that's why we can check it with other parameters. Since it is difficult to measure directly, so we go with other parameters. Now what are the other parameters through which we can measure the growth? So increase in number. increase in volume, increase in area, increase in length, increase in fresh weight, increase in dry weight. What are the other parameters through which we can measure the growth means? So we can tell it can be increase in fresh weight, right, or it can be increase in dry weight also, or it can be increase in length, it can be increase in length, it can be increase in area also, it can be increase in volume also. Finally, it can be increase in the number of cells also. So these are the other parameters which we use to measure the growth. For example, if we have to take an example for the increase in the number of cells, we can take the example of root maize plants, root apical meristem. If we take maize plants, root meristem cell, root apical meristem, you know how much it will divide. So in 1 hour it divides and it makes 17,500 cells per hour. That much fast it will divide. You cannot count 17,500 numbers also, but it can make 70,500 cells per hour. So the example is maize plants, root apical meristem. It divides it makes 17,500 cells per hour. So in this manner, this is an example. If we have to measure the size, if we have to measure the size or the area, we can take the example of watermelon. Watermelon increases its area or size up to 350 times. The cell which is this much, it will increase in its area up to 350 times. So that is measure in area or size. What example we have taken here in NCERT, they have mentioned about watermelon. Watermelon will increase in size by how much? 350,000 times. So for increase in size, watermelon is example. For increase in number, maize plant is an example they have given. For increase in length, if you see the pollen grain, when it is germinating, it will make a pollen tube. This pollen tube keeps on growing through the style till it reaches the ovary. Means here pollen tube growth is a measure of increase in length. For length, what example we can tell? Pollen tube growth is an example for increase in length we can tell. And for increase in surface area, we can take the example of expansion of a leaf, right? So for increase in, another example, surface area, it is expansion of the leaf. Now earlier the leaf is this much, now it has grown to this much. So increase in expansion of the leaf is an example of increase in surface area. So like this, since we cannot measure the protoplasm, we go with the other different parameters to measure the growth. Now coming to the phases of the growth, how many phases are there in the growth? First one is called meristematic phase, which is considered as an actively dividing phase. Second one is called as elongation phase, and the third one is called as maturation phase. So come on, tell me, what are the phases of growth? What are the phases of growth? For example, even in our life cycle also, so first we are born, from there till 18 years we keep growing. That is like growth phase for us. After that, what happens? Then we mature and we take a particular functions. Here also in plant we can tell there are 3 phases of growth. The first one, where it is actively dividing, is called meristematic phase. It is called meristematic phase. And the second phase, meristematic phase, what happens? Only the number of cells will increase. They divide and they increase the cells will be small, but in elongation phase, these cells will elongate. They become bigger. They reach their maximal size. So that phase is called as elongation phase. And the third phase, after reaching its particular dimensions, maximal size, then what it will do? It will get matured, it will get specialized, and it differentiates, takes up a function. That is called maturation phase. The third phase is called as maturation phase. So take a screenshot, then we will talk about the characteristics of meristematic phase, elongation phase, and maturation phase. Now let us see what are the characteristics of the cells which are in each phase. Now if it should be an actively dividing cell, if it should be an actively dividing cell, what should be the characteristics? So characteristics of meristem cells. Characteristics of meristematic cells. What are the characteristics of meristematic cells? Now the cell should have thin wall so that it can easily divide. The cell should have abundant cytoplasm. It should have prominent nucleus, right? Nucleus has to divide, so nucleus should be prominent. Cytoplasm only contains the energy, resources and all, reserve materials and all. Cytoplasm should be more. Nucleus should be prominent. The cell wall should have only primary wall, and they should be connected by plasmodesmatic connections. There should not be any free spaces like vacuoles and all, and it cannot store secondary metabolites like tannins, gums, resins, oils and all those things. So these are the characteristics of meristematic cells. What are the characteristics of meristematic cells? So meristematic cells will be small in size. They will be small in size and they have thin cellulosic wall only, cell wall. They have thin cellulosic cell wall which is called as a primary wall. Then we can tell that they have abundant cytoplasm. They have abundant cytoplasm, prominent nucleus, well visible nucleus. Now it will not give space for vacuole and all. There is no space for vacuole. Cell is very small, no? So no vacuoles. Rich of energy sources in the cytoplasm. It has to divide, no energy is required. Rich of energy in cytoplasm, and it will not store secondary metabolites. No storage of secondary metabolites. Now what do you mean by secondary metabolites? Like gums, resins, tannins, all those things. Gums, tannins, it can't store. So these are the characteristics of meristematic cells, and the meristematic cells are connected by plasmodesmatic connections. So they are connected by plasmodesmatal connections. So these are the characteristics of meristematic cell. Because it has to divide, the wall should be thin. Because it has to divide, nucleus should be there. Because it needs energy, cytoplasm should be there. It cannot accommodate vacuole, give spaces and all since the cell is small, and they should be interconnected by plasmodesmatal connections. Now let us see the second cell. What is the second zone? The second zone is called maturation, elongation zone. So what are the characteristics of the cells which are in the elongation zone? Now here in meristematic zone, we are telling this is the meristematic zone where the cells are very small. They are keeping on dividing, right? They are keeping on dividing. Now coming to the next phase, which is called as maturation phase, elongation phase. In elongation phase, what happens? The cells will elongate. See, this much cell has elongated and become this much. Another cell also got elongated. Another cell also got elongated. And again, here also the cell got elongated. The cells are elongating. How do they elongate that much? Means they will take vacuoles. The vacuoles will stretch and they help in expansion of the cells. Means the one important character of elongation phase is development of vacuole. So we can tell what are the characteristics. Development of vacuole will be developed, and then they make the secondary walls. Another characteristic we can tell, they make new walls. Means after primary wall, secondary wall will be formed. So new layers on cell wall will be formed. So these are the characteristics of elongation phase. Now let's go to the third phase. What is the third phase called? So it is the phase of maturation. Phase of maturation. Now in phase of maturation, what happens? So they have grown to their maximal sizes. Now they differentiate. They show some changes, right? So some changes in the protoplasm. For example, take our RBC. Our RBC at the time of maturation, it will lose nucleus so that it can provide space, so that it can take more oxygen, more nucleons in that space also. So that means it shows protoplasmic changes. Next, it develops walls also, and all this is called differentiation. Finally leads to maturation, and then it will take up a function. Now come on, tell me, what are the characteristics of maturation phase? They attain their maximal size, the wall becomes thick, and the protoplasmic modifications will be seen. So in the phase of maturation, we can tell the cells attain maximal size. They attain maximal size, and the cell wall shows thickenings. The cell wall will show thickenings, and the protoplasm shows modifications. The protoplasm shows changes. For example, if you take xylem, in xylem we have tracheids and we have vessels, right? So tracheids and vessels both are dead cells. If they are dead cells, they should not have protoplasm. They have to develop a lumen. That means when they are maturing at this time, they will lose their protoplasm. They develop a lumen, so, and they make thick lignin walls and suberin walls, so that they help in conduction. So these are the three phases of growth. Meristematic phase. This is meristematic phase. Next one is elongation phase. Above that will be maturation phase, where it will take up a function. Take a screenshot, then we will talk about growth rates. Now, what do you mean by growth rate? Rate means we are calculating it, we are measuring it. So increase in growth per unit time, per day how much it has grown, per month how much it has grown. So per time, if you are calculating, that is called as growth rate. Right? Come on, tell me, what is growth rate? It is increased growth per unit time. Increased growth per unit time. It is called as growth rate. How many means we are measuring it, means growth rate is expressed mathematically. There are two types of growth rates. One is arithmetic growth rate, and the second one is geometric growth rate. What are the two types of growth? There are two types of growth rates. First one is arithmetic growth, and the second one is geometric growth rate. The first one is arithmetic growth rate, and the second one is geometrical growth rate. Now, what happens in arithmetic growth rate? Now, the beautiful example for arithmetic growth rate is the division of the root cells. The root cells constantly they grow by showing mitotic divisions. The examples of arithmetic growth rate are, so constant growth of the root by mitotic divisions. By mitotic divisions is an example for arithmetic growth rate. Now in NCERT, they have shown us this picture. They have taken one cell. This is one cell. This cell is dividing by mitosis, and it makes two cells, right? It will divide by one mitosis. After mitosis, we will get two cells. The two cells are shown like this. Right? So this is one cell, and this is the other cell. This is one cell, and this is the other cell. So one cell, after one division, it became two cells. In these two cells, one cell will retain the ability to divide. The other cell will go to elongation phase and maturation phase. So think that this cell will retain, it keeps on dividing, will retain the ability to divide. This cell will retain the ability to divide, whereas the second cell, you know, so the second cell will develop. It will go to development. It will go to development, means it will go for elongation and maturation. It will go for elongation and maturation, means it will not divide further. Now next, if you see, since this cell is not dividing, then only this cell has retained ability to divide. So now again what happens? This cell will now divide into two cells. So then what do you get? So this cell will be like this only. Then we will get three cells, no? So the three cells are this cell will be like this only, but this cell got divided. First cell, second cell, third cell. Now again in these two cells, this cell has gone to elongation maturation. Now this cell also will go for elongation maturation. Only this cell will regain the ability to divide. Only this cell will regain the ability to divide. So again, from here, draw two arrows. This cell will be as it is. This cell will be as it is. So that means if you see the next line, one has divided into two cells. This cell will be like this. This cell will be, this is the third cell, this is the fourth cell, 1, 2, right? So now here three cells will go for elongation, only this cell will divide. So if you see in this manner, one cell became two cells became three cells became four cells. Again, if you have to continue, so it will be in this manner. It will be in this manner. So is there increase in number one by one? 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. One cell became two cells became three cells became four cells became five cells. So there is a linear increase in the number of the cells. Now if we have to plot the figure, now what should we take on x-axis and what should we take on y-axis? On x-axis we can take the time, on y-axis we'll take the length. We'll take the length. We are measuring here in terms of length. So here one cell, right? After some time 2 cells, after some time 3 cells, 4 cells, 5 cells, 6 cells, like that it will grow. Now when you combine all these dots, what shape graph do you get? A linear line we get, right? So we get a linear line. So that means for arithmetic growth, the plot is linear. So we get a linear curve. And you are measuring here in terms of length. So the formula will be LT is equal to, what will be the formula? The formula will be LT is equal to L0 plus RT. So LT, length of the plant after time T. So what is LT? The length of plant at time T. Then what is L0? Initial length. And what is R? R is growth rate. R, how much it has grown? One cell. And what is T? Time period. T is time period. So this is a formula for arithmetic growth rate. This is a curve for arithmetic growth rate, and this is the way the arithmetic growth happens. Take a screenshot. We will continue. We know growth can be measured by two types. One is arithmetic growth and the second one is geometric growth. Now see, looking at the pictures itself, we can tell that arithmetic growth means here one cell is there. In one cell has divided and it has made two cells. In these two cells, what happened? This cell will go for elongation and maturation. It will not divide further. Whereas only this cell keeps on dividing. You can see this line. Only this cell keeps on dividing. So that is why one cell will grow into two cells. It will divide into 3 cells, 4 cells, 5 cells. Constantly the cells are increasing. So there is not a jump in the increase in the number of cells. At a constant rate they are increasing. So it is called arithmetic growth. Now the formula for explaining arithmetic growth, the mathematical equation for arithmetic growth is LT is equal to L0 plus RT. Length at time T, initial length, R is growth rate, and T is the time period taken. And if we plot it, taking time on x-axis, height of the plant on y-axis, since it is growing 1, 2, 3, 4, 5, we will get a constant linear growth. And examples for arithmetic growth is root elongation at a constant rate. Root elongation at a constant rate, shoot elongation at a constant rate can be examples for arithmetic growth we discussed earlier also. Now coming to geometric growth. In geometric growth, one cell has divided into two cells. This is the first cell and this is the second cell. Here both the cells are retaining the ability to divide. Since both the cells are retaining the ability to divide, so this cell will again make two cells. This cell also will make two cells. So then it's multiplying, right? 2 became 4. Now 1, 2, 3, 4. All the 4 cells have regained the ability to divide. So then this will make 2, 2, 2, 2, right? So 2, 4, 6, 8. So it's multiplying. So that's why we call it exponential. At an exponential range they are increasing. 1 will become 2 cells, 2 will make 4 cells, 4 will make 8 cells, 8 to 16, 16 to 32, 32 to 64, like that they are multiplying. So when we plot it by taking time on x-axis and here the size or weight of the organ and y-axis, then which shape of the graph we are getting means sigmoidal curve we are getting. So in sigmoidal curve there are 3 parts. This is the first part, then this is the next part, and this is the third part. Now first part where it has slowed down the growth means it is getting established to the changed environment. The growth is very slow. That period of growth is called lag phase. Now next, when it gets established, when it starts consuming the nutrients and it starts multiplying, right? It starts multiplying to the power when it multiplying, then it peak increases the growth rate. That's called exponential phase, or we can also call it as log phase. This is lag phase where it has slowed down. Log phase it has increased. So that's what it's called exponential phase, where it's fastly growing, which is active. Now the third one is stationary phase, where the nutrients gets exhausted. The nutrients what you have supplied gets exhausted. Since the nutrients gets exhausted, it cannot grow further. It maintains its growth, right? It retains as it is. That is called stationary phase. So which shape are we getting here? Sigmoidal growth curve we are getting here. Since we are measuring here the weight, the mathematical formula for geometric growth curve goes with W. Final weight is equal to initial weight E power RT. What is E? Base of natural logarithm. R is growth rate and T is the time period taken. So the formula for geometric growth rate is W1 is equal to W0 E power RT. Initial weight, final weight, base of natural logarithm, growth rate and time period taken. Examples for geometric growth rate if you take. Now higher plants exhibit this type of growth only, even we. So when we are born we are this much, then we keep on growing. After reaching this much height we stop further growth. That means we are also showing this phase of growth only. So growth of an organism in a natural condition, right? Resources will get exhausted at one point of time, then the growth gets maintained off. So growth of organisms at natural conditions is this type of growth only. Now typical growth of a cell or a tissue or an organ if you take, it will be like this only. It will grow very fast. After reaching its particular size it stops its growth. Next one, bacteria growing in a petri dish in nutrient medium is also an example of geometric growth only. And plants which are showing seasonal activity, in one particular season they grow fast, they flower and they fruit, right? So mango and all, other plants and all. So the plants which are showing seasonal activities or the trees which are showing seasonal activities also exhibit sigmoidal growth curve, means geometrical growth curve only they will show. Now this is the difference between arithmetic and geometric growth curve. Now in NCERT, after these two pictures, they have given the picture of a diagram and they showed us. If we have to see the zygote, this is zygote. Zygote divides into 2 cells first. So this has divided into 2 cells. Then it has divided into 4 cells. Then it will divide into 8 cells. If I have to go, these 4 cells will become 8 cells. Now 1 became 2, 2 became 4, 4 became 8. So this is geometric, means initial stages of division of the zygote development are geometric. Later on it becomes arithmetic. Later on it becomes arithmetic. When we see, when it is growing like this, then they will show us. So at this pace it is showing geometric growth, but here it is showing arithmetic growth. Here it is showing arithmetic growth and here it is showing geometric growth they have given. So the initial phases are geometric only since the number is increasing, but later on it shows both geometric and arithmetic growth rate. So understood till here the types of growth rates. What is growth rate? Growth rate means increase in growth per unit time. So we have taken time and we have calculated arithmetic growth rate and geometric growth rate. Growth is qualitative or quantitative? Growth is quantitative. So we can quantitatively compare the growth in 2 types also, which is called as absolute growth rate and relative growth rate. Take a screenshot. We will talk about the quantitative comparisons of the growth in terms of absolute growth rate and relative growth rate. We have seen what is arithmetic growth and geometric growth. Growth can also be measured by quantitative comparison. If we have to measure growth by quantitative comparison, then there are two techniques. One technique is, it's called as absolute growth rate determination, and the second one is called as relative growth rate determination. So there are two methods for quantitative comparison of the growth. What are the two methods? One is absolute growth rate and second one is relative growth rate. Now let us see what is absolute growth rate and relative growth rate, and through these leaves we will find out what are the absolute and relative growth rates. Now see here, the definition of absolute growth rate is given like this in NCERT. It is the measurement and comparison of the total growth. Take this leaf, so you measure it. You measure it and you compare it. What is its initial size and how much it has grown. So measure it. So you have measured, it has grown 10 centimeters. You measure it and you compare it. Its initial size is 5 centimeters. So measurement and comparison of total growth per unit time. Growth rate means it is increase in growth per unit time, per time, 1 day now, 1 week, like that. So measurement and comparison of total growth per unit time is called as absolute growth rate. Then how can you give it mathematically? Absolute growth rate is equal to final size. Here the final size is 10 centimeters minus initial size. The initial size is 5 centimeters by time, right? By time, per unit time means we have given by time. Okay, so this is a formula which goes with absolute growth rate, right? Now let us see, like this leaf is showing how much absolute growth rate and this leaf is showing how much absolute growth rate. Now we'll apply this formula. Now absolute growth rate of this leaf. Okay, so if we have to take the leaf A, absolute growth rate of A. So what is the final size? The final size is 10 centimeters. So 10 minus, what is the initial size? Initial size is 5 centimeters by time, 1 hour or 1 day. So then how much do we get? So we get it as 5 centimeter square. So how much are we getting? We are getting 5 centimeter square per hour. Per hour. This is the absolute growth rate of this leaf. In the similar manner, let us go for the second leaf B. Absolute growth rate of the next leaf B. Now here, what is the final size? The final size is 55 centimeters they have given in NCERT. So 55. And what is the initial size? Here the initial size is mentioned as 50 centimeters. So I took the final size as 55 minus initial size. Initial size is 50 by 1 hour. So here also 55 minus 50 gives you 5 centimeters square per hour only. Means leaf A also and leaf B also. In this term, in absolute growth rate, both are showing 5 centimeters square per hour only. Both are showing 5 centimeters square per hour only. Now let us see what is the definition for relative growth rate. In NCERT, the definition goes like the growth of a given system per unit time expressed on a common basis. Any system, leaf or root or stem, the growth of a unit, the growth of a given system per unit time. Per unit time, you are taking time, per time only, per time expressed on a common basis. What do you mean by common basis? Here you are measuring the size. Now what is the formula for relative growth rate? Relative growth rate is equal to the same thing again, final size. Final size is 55 minus initial size. Initial size is 50 centimeters by the initial size expressed on a common basis, expressed per unit size they are telling, right? Per initial parameter, per initial size. What is the initial size? The initial size is 50 here. The initial size is 5 here. So by initial size again we have to give, and per time. Per time means 1 hour if you calculate. So this is the definition for relative growth rate. Now let us see, find out the relative growth rate for this leaf and the relative growth rate for this leaf. Okay. Now let's go. Relative growth rate for leaf A. Relative growth rate for leaf A. What is the final size here? A bar, right? It is A bar. Now how much it is A bar? Final size is 10 centimeters and minus initial size. Initial size is 5 centimeters by initial size. Initial size is 5 into time. Time is 1 hour into 100. So this is a formula. 10 minus 5, 5. 5 by 5, right? So how much you do get? 5 by 5. If we get 5 by 5 into 1 by 1 into 100, so they will go off. That means 100% you're getting. That means this leaf is showing 100% relative growth rate. So the relative growth rate of this leaf A is 100%. Okay. Now in the same manner, let us calculate for B also the relative growth rate for this leaf called B. Now the formula is final size. The final size is 55 centimeters they told. It is 55 centimeters minus initial size. The initial size is 50 centimeters by the initial size again. Again the initial size is 50 into 1 by 1 into 100. Now if we take 55 minus 50, it's 5. Again here how much do you get? Here you get it as 5 by 50. 5 by 50 into 1 by 1 into 100. Then here if you see like 5 in 10 times it will go, right? So it is 10 times. So that means the relative growth rate of B is 10%, whereas the relative growth rate of A is 100%. Correct only. See, 5 centimeters has doubled by another 5 centimeters. It became 10 centimeters. Means 100%. 5 another 5 got increased, it doubled, so that is why it's 100%. Whereas see, 50 centimeters did not double and become 100 centimeters, it just increased by 5 centimeters and it became 55 centimeters. Means relative growth rate of this one is 10%, relative growth rate of this one is 100%, whereas absolute growth rate of both the leaves is the same 5 centimeters per hour. In NCERT they have asked you relatively which leaf has shown greater growth rate. Means come on, tell me, this one has shown relative growth rate, this one has shown relative growth rate, or this one has shown relative growth rate. We can tell this leaf is showing lesser relative growth rate and A is showing greater relative growth rate. So A shows greater relative growth rate than B. This leaf if you consider it as A and this leaf if you consider it as B, A is showing relatively more growth rate. How much growth rate? 100% than B. So B is showing only 10%. Take a screenshot, then we will talk about the conditions of the growth. Now, what are the requirements we require for growth? Now, come on, tell me, if the growth has to happen, oxygen is it required? Water is it required? Nutrients are they required? So, not only that, we can tell temperature is also important for growth. Gravitational force is also important. Environmental factors are also responsible for growth. Now, if you are asking me what are the conditions for growth, means there are so many conditions. But what are the important conditions of growth? So, if we write on the next topic in NCERT is conditions of growth. Whether you believe or you don't believe, you have to read NCERT textbook in this manner only if you want to score out in NEET UG examination. If you have to get 360, you have to read NCERT in this manner, line to line, point to point, then only it is possible for you to score. Now, conditions for growth. In NCERT, they told water is required for growth, oxygen is also required for growth, nutrients, macronutrients and micronutrients are also very essential for the growth, and they have given explanation points also, that also we'll see. What are very essential conditions for growth? So we can tell water is one, oxygen is another one, and nutrients. These three are very essential. They are very essential for growth. Now I told you, I'll tell you the points also, why water is essential, why oxygen is essential. Come on, if we go with the first point, water. Now plant, the cells will divide. After dividing, they'll go to the next phase, which is called as enlargement phase. Means if the cell is enlarging, how does it enlarge? It will develop a vacuole. In that vacuole, it will store water and it enlarges. Means plant grows in size by cell enlargement. So we are talking about water now. We are telling plant grows in size. It is growing in size. How does it grow in size? From meristematic zone, it's going to elongation zone. It is enlarging zone where it will develop a vacuole. In that vacuole, it starts accumulating the water. So plant grows in size by, we are telling it is cell enlargement. During cell enlargement, it will generate or it will make vacuole and in that vacuole it accumulates water and other substances. If it accumulates water and other substances, what will happen to the cell? The cell exhibits turgidity. Because of that turgidity, the cell will show extension, which is called as growth. Because of accumulation of water and other substances in this vacuole, the cell will exhibit, cell shows turgidity. Now this turgidity is responsible, it is responsible for extension of the growth. This turgidity is responsible for extension of the cell, or we tell that the cell is growing. So we are telling the cell will enlarge in its size by developing a vacuole. In that vacuole it keeps water, it keeps substances, and it exhibits turgidity, which leads to extension, right? Not only that, that means we can tell that plant growth is very much related to the water status, right? So that means plant growth is intimately linked to the water status. So hence we can tell, hence basing on this point we can tell plant growth is intimately associated. Plant growth is intimately associated with what? With the water status. It is intimately associated with the water status. Not only that, water provides the medium for the enzymes to act, right? So water, 50 to 70% of the cell is made up of water only. In that water only, in that protoplasm only, all the enzymatic reactions will happen. So we can tell that water also provides the medium for enzymatic activities which are needed for the growth. So we can tell the second point that water also provides the medium. Water will make the cytoplasm. In that cytoplasm only all the enzymatic activities will be going on. Water also provides the medium for enzymatic activities needed for the growth. For the enzymatic activities. For the enzymatic activities we are telling needed for the growth. So these points are the sufficient. Tell that water is a condition required for growth. Now let us talk about oxygen. So before this chapter we have respiration chapter. In that respiration chapter we'll study aerobic respiration. If a nutrient undergoes oxidation in presence of oxygen, it will give 38 molecules of energy. One glucose molecule, when it undergoes complete oxidation, gives 38 molecules of ATP. That means oxygen, what it will do? Oxygen, it helps in releasing the metabolic energy. It will do metabolism, it will release the energy. In presence of oxygen, energy is more. In absence of oxygen, energy will be very less. So oxygen releases metabolic energy required for the growth. Oxygen releases metabolic energy. In the presence of oxygen, so much amount of energy is released. This energy is required for the growth. It releases metabolic energy which is required for growth. Right? So second factor also got over. Now let us talk about the third factor. What is that? Nutrients. Which type of nutrients are there? Macronutrients are also there, micronutrients also there. What they will do? They will synthesize the components of the protoplasm and thus they help in the growth. So coming to the third factor, nutrients. How many types of nutrients are there? Macronutrients and micronutrients. Macronutrients and micronutrients. What do these nutrients do? They will synthesize the protoplasm and it helps in the cell growth. They will synthesize the protoplasmic content, and thus they also contribute to the growth of the plant, right? So these are the very essential factors: oxygen, water and nutrients. Not only that, I told you temperature, optimal temperature is very much required for the plant growth, and if you increase the temperature where plant cannot tolerate, it is deleterious and the plant will die. Not only that, environmental signals also help in the growth of the plant. What environmental signals? Means its response to light, response to gravity also will affect the growth. So these are three main factors. One is water, other one is oxygen, other one is nutrients. Apart from that, we can tell optimal temperature also, and then we can tell environmental signals also. Environmental signals means response to light, response to growth will also affect the growth. They will also affect the growth. Children, these are the points which they have mentioned on the side heading, conditions for growth. Understood? 3 main essential conditions they told, and other 3 conditions are temperature, environmental signals. Now the next topic in NCERT is what is differentiation, what is dedifferentiation, what is redifferentiation. Take a screenshot, then we will move on to the next topic. Right, so what is the next topic? It is what do you mean by differentiation, dedifferentiation and redifferentiation. We'll see that now. Now all the plant cells will come from the actively dividing cell, which is called meristematic cell. Now this meristematic cell, it will divide and then it will undergo enlargement and then it will mature, right? So let us see what are the meanings of differentiation. Next one, dedifferentiation, and the third one, redifferentiation. Right, children? Now let us see what is differentiation, dedifferentiation and redifferentiation. Now meristematic cell, the actively dividing cell, it divides, then it enlarges, then it matures and then it will take up a function. That process is called differentiation. What is differentiation? Meristematic cell. Meristematic cell developing into a permanent cell. If the meristematic cell enlarges, reaches its maximum size, gets matured, takes up a function, then that is called as permanent cell. What's the difference between meristematic cell and permanent cell? Meristematic cell will divide, but the permanent cell has lost the ability to divide and it is taking up a special role, specific role, right? So now, if we have to see what are the meristematic cells which have developed into permanent cells, if you have to see the examples, understood, children? Meristematic cell converting into a permanent cell like parenchyma, chorenchyma, sclerenchyma, xylem, phloem, all these are permanent cells. This process is called differentiation. Examples: root apical meristem you take, shoot apical meristem you take, and cambium you take. What are the examples of the meristematic cells becoming permanent? In NCERT, they have mentioned root apical meristem. Then they also told shoot apical meristem. Then they also told the cambium. This root apical meristem, shoot apical meristem and cambium. So they are meristematic cells. They differentiate. What do? They will expand, they will mature, means they will differentiate, they will mature and they will take up a function. So all these 3, they will differentiate. They will differentiate, they will mature and they take up the function. Now root apical meristem will make the root, the parts of the root. What will root apical meristem do? Root apical meristem will make the root. Now what is the function of the root? Absorption. Now shoot apical meristem, what it will do? It will make shoot. What is the function of the shoot? To conduct water from the root to the upper parts, conduction. And what will this cambium do? The cambium will make the vascular tissue. Cambium will make the vascular tissue. What are the examples of vascular tissue? Xylem and phloem. What will xylem do? Helps in water transport. What will phloem do? Food transport. right? Food transport. So meristematic cell becoming permanent. Examples for meristematic cells: root apical meristem, shoot apical meristem, and cambium. Root apical meristem will make the root. Now root is differentiated. Epidermis, cortex, all those regions are differentiated. It's doing a function. What is it? Absorption of water and minerals from the soil. Now shoot apical meristem will make the shoot. What shoot will do? Shoot is a conductive tissue which helps in conduction of water and minerals from the root to the upper parts, conduction of food from the leaves to the lower parts. Now cambium, it will make a vascular tissue called xylem and phloem. Xylem helps in transport of water, phloem helps in transport of food. Right children? Understood till here? So this is differentiation. This is differentiation. Now when a meristematic cell, which is a small cell which has prominent cytoplasm, it has abundant cytoplasm and it has prominent nucleus, it has thin wall like this. When it is undergoing, when it is becoming permanent cell, when a meristematic cell will become permanent cell, there are so many changes to be noticed. There are so many changes. What are those changes? Changes we can see in the cell wall also. Changes will be in the protoplasm also. We can see the changes in the wall deposition also. We can see the changes in the inside content protoplasm also. For example, we'll take tracheary elements, means xylem, tracheids and vessels. In that, what happens during differentiation? So if we take tracheary elements, right? So what do you mean by tracheary elements? It includes tracheids and vessels. It includes tracheids and vessels. Now these tracheids and vessels, when they are becoming permanent tracheids and vessels, now what changes they show? You can see that the cell has elongated. So during differentiation, it shows changes. You can see the cell got elongated. Not only that, it shows depositions, lignin depositions it will show on the wall. Means secondary wall gets deposited. This is another change we can see. And the protoplasm here will disappear. Meristematic cell is a living cell, whereas tracheids and vessels are dead cells. Since they are dead cells, the protoplasm will disappear. Nothing will be there inside. We call it as a lumen. We call it as a lumen. Nothing will be there inside. And this is lignocellulose wall. Since there is nothing, it can easily help in water conduction. Since there is nothing, it can, from the lumen, it can easily help in water conduction. Because of the secondary depositions of lignin and cellulose, now it can retain that turgidity, it can retain that elasticity, it can retain that tension and conduct the water from the roots to the tall trees also. So during differentiation, so it shows major structural changes. Structural changes on the wall also, structural changes in the protoplasm also. We took tracheids as an example to understand. So children, all this is about differentiation. Now let us talk about what is dedifferentiation. We finished differentiation. Now let us talk about what is dedifferentiation. Meristematic cell becoming permanent cell is differentiation. Meristematic cell means actively dividing cell. Permanent cell means the cell which has lost the ability to divide. It forgot the ability to divide. It has matured and it is specialized in taking up a function under certain conditions. For example, this is your skin. It will not divide. But if any damage happens, if any wound happens, when the skin is removed off, the new skin will be made. No, in the same manner, in the plant also, under certain conditions, the permanent cells which have lost the ability to divide will regain the ability to divide. Okay, that means meristematic cell becoming permanent is differentiation. Permanent cell becoming meristematic is dedifferentiation. What is dedifferentiation? PC means permanent cell. Becoming MC means meristematic cell. The permanent cell becoming meristematic cell. Let me write down. The permanent cell becoming meristematic cell again. The permanent cell which has lost the ability to divide, under certain circumstances it will regain the ability to divide and again it becomes actively dividing cell. Then that process is called as dedifferentiation. If we have to take the examples of dedifferentiation, what are the examples of dedifferentiation? Dedifferentiation means interfascicular cambium formation. Another example is cork cambium formation. Now intervascular cambium and cork cambium, both of them will be formed from a simple tissue called parenchyma tissue. Parenchyma tissue, what is it? It is a permanent cell. It is a permanent tissue. It has lost the ability to divide, but it has regained the ability to divide, and it again became meristematic. Now it is intravascular cambium which is meristematic, cork cambium which is meristematic. This process of permanent cell becoming meristematic is dedifferentiation. Got it? Examples are intervascular cambium and cork cambium. Now let's go to redifferentiation. Let us see, we finished here dedifferentiation. Now let us see what is redifferentiation. What is redifferentiation? Meristematic cell becoming permanent is differentiation. Permanent cell becoming meristematic is dedifferentiation. Now what is redifferentiation means? This meristematic cell again becomes permanent. It again becomes permanent. Means, can we tell that it is differentiation of dedifferentiated tissue? What is redifferentiation? It is differentiation of redifferentiated tissue. It is differentiation of redifferentiated tissue. Now see here, meristematic cell, permanent cell, differentiation. Permanent cell became meristematic, dedifferentiation. Now this meristematic cell again becomes permanent. This meristematic cell again becomes permanent. It is redifferentiation. So I told it is the differentiation of dedifferentiated tissue, not re. It is the differentiation of dedifferentiated tissue. Differentiation of dedifferentiated tissue is called redifferentiation. So what is dedifferentiated tissue? Vascular cambium, cork cambium. Now from vascular cambium, what do we get? From cork cambium, what do we get? They will be the examples of redifferentiated tissue. For example, if you take vascular cambium means it is vascular cambium. Come on, what do you get from this vascular cambium? What do you get from cork cambium? They are asking. Now this is parenchyma, this is meristematic cell. If this vascular cambium cuts out and in, it will make secondary xylem, secondary phloem. From vascular cambium we get dedifferentiated tissues like secondary xylem and secondary phloem. In the same manner, from cork cambium we get cork and secondary cortex. From cork cambium, which type of redifferentiated tissues we get? We get cork and secondary cortex. So this we studied it in 6th chapter, anatomy of flowering plants. There we studied this in detail. So I think we understood what is differentiation, what is dedifferentiation, what is redifferentiation. Examples of differentiated tissue: root, stem, leaf and all those things. Examples of dedifferentiated tissue: vascular cambium and cork cambium. Examples of redifferentiated tissue: secondary xylem, secondary phloem, cork and secondary cortex. In NCERT they have asked a question: in plant tissue culture, you will take an explant and we will grow it. Now the explant, which can be a root, stem or leaf, which is a permanent tissue. Now after few days it will grow into a white amorphous undifferentiated mass of tissue which is called as callus. Now what is this called as? They asked in plant tissue culture. Now getting callus is which process? So I told from leaf you got, leaf is a differentiated tissue. So what is that process called? Dedifferentiation. Leaf, which is a permanent tissue, became meristematic again, formed callus again. So in NCERT they asked in plant tissue culture, formation of callus is an example for dedifferentiation. Formation of callus is an example for, I will tell, it is dedifferentiation. Why are we telling it as dedifferentiation? Because in plant tissue culture you have taken leaf. Leaf is a permanent tissue. It has developed into a white undifferentiated amorphous mass. So became meristematic again. So it will be an example of dedifferentiation. Take a screenshot short. Then we will talk about the next one which is there in NCERT, that is development. So the next topic is development. Now we told development is a sum total of two processes, growth and differentiation. Growth, we have seen, growth is irreversible, it is permanent, it is conspicuous, it is visible. We told all those things. Now differentiation, we told what is differentiation, dedifferentiation, redifferentiation. With examples we have seen. Now let us talk what is development. That's what the chapter is about. Now development is a sum total of two processes. What is that? Growth and differentiation, right? So development is the sum total of two processes. What are they? First one is growth and the second one is differentiation. So we have seen this. What is growth? What is differentiation? We have seen. Now plant development, it starts from a seedling, right? So from where does it start? It starts from a seed. Now seed develops into seedling. Seedling will develop into a plantlet. Plant grows into a matured plant or a tree. Matured plant, or it will become a tree. Then it undergoes senescence, old age, then finally death. Then afterwards it shows senescence, leading to the death of the plant. So are these the developmental stages? Now seed develops into seedling. Seedling will grow into plantlet. Plantlet will mature enough. It's called as a matured plant or a matured tissue, matured tree. Then after some time it undergoes old age, which is called as senescence, finally leading to the death of the tissue. Now in NCERT they have given the diagrammatic representation. Now to explain what is development, let us see that. They started with meristematic cell. They started with meristematic cell. Now what is meristematic cell? Actively dividing cell. Now what happens to the meristematic cell? Meristematic cell will grow and it shows growth, right? It expands. What is that growth called? Plasmatic growth. Protoplasm will increase. So we have to call that as plasm increasing. So plasmatic growth. After exhibiting plasmatic growth, then what it will do? It shows differentiation. It gets differentiated to become a mature tissue. Meristematic cell, which is an actively dividing cell, it will grow, it exhibits plasmatic growth, then it shows differentiation. Then it shows differentiation and it becomes a matured tissue. So this is a dividing tissue, that is a tissue which has lost, right? Now plasmatic growth, can we call it as expansion? Differentiation, can we call it as maturation? Yes. In plasmatic growth what happens means expansion will happen. In differentiation, what happens in differentiation means maturation will happen. Now the mature tissue, it will grow. The matured plant, it will grow, it will undergo senescence. So the next stage is, we can write senescence. And senescence will lead to death. Senescence will lead to death. Sometimes we have seen arithmetic growth and geometric growth. In geometric growth, one cell will divide into two cells and both the cells will retain meristematic activity. Two cells will become 4, 4 will become 8, 8 will become 16. Exponentially, logarithmically they are increasing. So in that case, what happens? This meristematic cell, after it divides, now it will again show cell division, no? It will again come back like this, right? So where it is in geometric growth. Now what is that called? Cell division. It has divided and again came back, again that will divide. So we can tell this is called as cell division, right? So we have to call it as they are coming back and becoming meristematic again so that they can divide again. So this is the process of development. They will give you this picture and they will ask you A, B, C, D, E, F. Identify them. A is a meristematic cell, actively dividing cell. B, it is increasing its protoplasm, plasmatic growth. After increasing it to its maximal size, then it will undergo differentiation. It will become a mature cell. Now, increasing plasmatic growth is called expansion of the size. Now, differentiation process called maturation. From matured plant, it will go old age, then it will die. If it is a geometric growth, then the cells keep on dividing. They will become meristematic again. So, they divide and they become meristematic cell again. Understood? This is about development. Our next concept is plasticity. Take a screenshot. We will continue about plasticity. Right. What is plasticity? How many types of plasticities are there? It is the response of the plant. What is plasticity? It is the ability of the plant to show different kinds. Ability of the plant to form different kinds of structures. Plasticity means it refers to the ability of the plant to form different kinds of structures. It forms different kinds of structures during development, or it forms different types of structures during different habitats, during development or during different habitats. Right. Now, let us understand this clearly. Plasticity means it is an ability of the plant to form different kinds of structures in different stages of their life or in different habitats. Now, if it is showing differences in the development, what is that called? Developmental plasticity. If it shows differences in the habitat, on land it is like one way, in water it is like one way, what is that called? Environmental plasticity. So, during development, if it shows changes, that's called developmental plasticity. This refers to developmental plasticity, whereas this refers to different habitats, right? This refers to environmental plasticity. Environmental plasticity. I'll give the examples. Examples for environmental plasticity is buttercup. Right? Now this buttercup plant is called ranunculus. What is the other name of the buttercup? It is also called ranunculus. Now this ranunculus plant, now some parts of the plant are inside the water, some plants are aerial. If it is an aerial plant, how does the leaf look like? If it is an submerged plant, how does the leaf look like? If it is an aerial plant, then the leaf will have, right, broad structures like this. So if it is in aerial habitat, but whereas if it is in the aquatic habitat, to tide over that water waves and all, even the leaf will be in a split manner. So see, the leaf is in this manner when it is in water, so that water currents cannot break the leaf because it's already a broken leaf. Now in aquatic condition, the leaf is like this so that it will not tear, it's already torn. Now in aerial habitat, the leaf will be a proper broad leaf like this. What is this called? Environmental plasticity. Example, buttercup, which is also called ranunculus. Now come to developmental plasticity. Developmental plasticity is exhibited in which plants? It is exhibited in cotton, coriander and larkspur. Developmental plasticity examples are cotton, gossypia means cotton, coriander, and the third one, larkspur. Okay, the third one is called larkspur. Its name is delphinium. Larkspur is also called sometimes we remember it as larkspur, but in the paper they have asked delphinium. So larkspur is called delphinium. Now what do you mean by developmental plasticity? The juvenile leaf will be in different appearance and the adult leaf will be in different appearance. So if we see the juvenile leaf, how does it appear? If we see the adult leaf, how does it appear? Since you are telling it is developmental, in juvenile development the leaf is in different manner, in adult leaf it is in different manner. In juvenile the leaf will have small lobes like this. Now if you take the adult leaf will have prompt structures. It has nicely grown, right? So this is a, see, coriander we eat, right? The adult leaf will be like this, but the juvenile leaves will be very small. It will be like this. Now what is this plasticity called? Developmental plasticity. Examples: cotton, coriander, delphinium, larkspur. Now environmental plasticity: in the water it is like this, on the land it is like this. Example: buttercup or ranunculus. Take a screenshot, we will continue. We have seen what is growth and what is development. Now let us see, now growth and development are under control of which factors. So here they have given development of a plant is under the control of certain factors. Internal factors are called as intrinsic factors. External factors are called as extrinsic factors. Now what are the examples of internal factors and what are the examples of external factors? Now internal factors, if they are within the cell, then that is called intracellular factor. Who is there within the cell? Who will control the cell? Genes, right? So that means they are genetic factors. Now who is there outside the cell? Who can control the cell? Chemicals. So intercellular means chemicals, intracellular means genetic factors. Now examples for those chemicals are called as plant hormones, plant growth regulators they are. Okay, that only we are going to see in detail. First let us finish off what are the external factors, then we will come to plant growth regulators. Now what are external factors? External factors means plant growth is affected by water, it's affected by oxygen, it's affected by temperature also, it's affected by nutrition also. So all of them which are environmental factors, they come under extrinsic factors like light, temperature, oxygen, water and nutrition. Right children, understood till here? Now I told you as from now onwards we are going to talk about the hormones, plant growth hormones. They are also called plant growth regulators. What are the characteristics? Now plant growth regulator, first let us see what are the synonyms. What are the other names we can use it in literature? So one we wrote, it is called plant growth regulator, the first one. It is also called plant growth substance, second name. It is also called plant hormone, third name. It is also called phytohormone, fourth name. So we can give four names here. What are they? PGR, plant growth regulators, plant growth substances, plant hormones, phytohormones. These are the names, right? Now what are the characteristics? Like human hormones, are they big or very small? They are small. Like human hormones, they are complex in structure or simple in structure? They are simple in structure. Like human hormones, they are more in number or less in number? Less in number, right? And they are less in number, they are simple in structure and they are small in thing, but they are diversified in composition, chemical composition. Our hormones, if you take, either they are proteinaceous hormones or they are steroid hormones, fatty hormones, right? But here there are only 5 hormones, 5 plant hormones, but each hormone is made from separate chemical. Plant hormones are diversified in their chemical composition. So what are the characteristics? They are simple in structure, they are small, they are simple molecules, but they are diversified in their composition, chemical composition. I wrote here first, second, third, fourth, fifth hormone. So indole compound, from indole compounds auxin is made. The chemical name of auxin is IAA, indole acetic acid. Where is the indole acetic acid attached? To the third carbon. So it's called indole 3 acetic acid, which is a chemical name of auxin, which is derived from indole chemical compound. Next one, a purine is there, which is called adenine. From adenine, the second plant growth hormone is made. It is called as kinetin. So from adenine derivative, kinetin, cytokinin is made, and which adenine it is? At N6th position, furfuryl aminopurine it is. So the complete chemical name is N6 furfuryl aminopurine is kinetin. The third hormone, so if it is made from terpene chemical compound, it is called gibberellic acid. Gibberellic acid 1 to 100 are there, but the popular gibberellic acid is Ga3. The widely studied gibberellic acid Ga3. The first isolate is also Ga3, right? Then if the chemical composition, if it is a carotenoid derivative, then that is called as ABA, abscisic acid, which is another name of plant hormone. The last hormone, which is a simple gaseous volatile hormone, its name is ethylene. There are 5 hormones as I told you, but each hormone is having its own completely different chemical composition. Indole compound, auxin. Next one, adenine compound, kinetin. Then third one, carotene compound, if you take, it is abscisic acid. Terpene compound, if you take, gibberellic acid. If it is simple gaseous volatile compound, it is ethylene, right? So we can also classify plant growth regulators on the basis of their functions, whether they are having promoting roles or they are having inhibiting roles. If they are having promoting roles, then they are called plant growth promoters. If they have inhibiting roles, then they are called plant growth inhibitors. Now in these 5 hormones, auxin, kinetin, gibberellin, abscisic acid and ethylene, how many are promoters? How many are promoters? First, second, third are promoters. So auxin, cytokinin, gibberellic acid are promoters. Example for inhibitor, the 4th hormone is inhibitor. It's called as stress hormone. It stops the activity, stops the metabolism, slows down the metabolism, causes dormancy, all inhibiting actions it has. So under inhibiting hormone, ABA will fit. 5th hormone, ethylene, which is a simple volatile gaseous hormone, can neither fit under this category or this category, because it has some promoting activities, it has some inhibiting categories. So you cannot keep here because it's not a purely promoter. You cannot keep there because it's not purely an inhibitor. But then if you see which actions it is having more, promoting actions are, inhibiting actions are, largely it has inhibiting actions only. So take a screenshot, then we will continue with the characteristics of or the discoveries of the plant hormones. Right, now let us talk about the discoveries of plant growth hormones. You know, almost all the scientific discoveries are incidental, they are accidental only. So means intendedly they did not start working on that and they discovered it. So they were working on some other thing, they ended up with this discovery. So like that, all the 5 hormones, you know, plant growth hormones, their discoveries are accidental only. So we can write down the discoveries of all the 5 hormones are accidental. Discoveries of all 5 plant growth regulators are accidental only. All the 5 are accidental only. We will see one by one. Let us start with the first hormone, auxin. Right, let us start with the first hormone, auxin. You know, father's name, the scientist father's name, we can write it as father name is Charles Darwin and his son is called Francis Darwin. In the year 1881 they worked out. So in 1881, what is the name of the father? Charles Darwin. And son, now what is the name of the son? Francis Darwin. Now these 2 people, what did they do? So they were working on a plant. The name of the plant is canary grass. So they observed in which plant? In canary grass. Now this is a common name. Sometimes if they want to make the question difficult, they can ask you what's the scientific name. With the scientific name also they can ask you. The scientific name of canary grass is Phalaris canariensis. What is the scientific name of canary grass? Canariensis is the scientific name of canary grass. Now what did they observe? In NCERT diagram they have given that the canary grass coleoptile. This is like this, and we are subjecting unilateral illumination of light. So when you subject unilateral illumination of light, so this is canary grass. This is the shoot tip of the canary grass. Shoot tip, what is it called? Coleoptile. This coleoptile started bending towards the direction of the light. Means it started bending. Okay, so why, what happened? Means at this area auxin is synthesized. Now this auxin moves from the this lighted part to the shaded part. It means auxin is moving from this side where light is there to the other side. Here dark is there. The auxin is moving here. Since auxin is moving here, since auxin is a growth promoting hormone, what the auxin will do? Auxin will help these cells to grow bigger. It is helping these cells to grow bigger. So these are bigger cells. Whereas this side where auxin is not there, these cells are quite smaller. Means the growth on this side is less, whereas the growth on this side is more because of accumulation of auxin. So this response of the auxin movement from the light side to the dark side and making the plant to bend in the direction of light is called phototropism. What is this called? Phototropism. This was the observation made by father and son Charles Darwin and Francis Darwin. Later, another scientist, his name is F.W. Owen. He worked on which plant? He worked on oat plant. Now, what is the scientific name of oat? It is called Avena sativa. So, Went worked on Avena sativa and he isolated the compound. He isolated auxin from the tips of Avena plant. So Went, what did he do? He isolated auxin from the tips of Avena plant. So Went isolated the auxin from the tips of Avena plant. So this is called as a bioassay test for auxin. If they ask you what is a bioassay test for auxin, it is Avena curvature test. Note down this. So what is the bioassay test for auxin? It is the test which is done by Went only. Went isolated it, he named it, he isolated it from the Avena tips. So the bioassay test for auxin is Avena curvature test. It is called as Avena curvature test. The Avena plant also, it bended in the same direction. In this manner, auxin has been identified. Take a screenshot, then we will talk about the second hormone, which is called as gibberellic acid. Yeah, now let us talk about the second hormone. We finished auxin. Auxin was discovered by father, his name is Charles Darwin, son Francis Darwin. Now let's talk about the second hormone. I told the second hormone is gibberellin. Okay, it is called gibberellin. Now there is a disease. What is the name of the disease? BOTANY disease. What is the name of the disease? BOTANY disease. It is also called foolish seedling disease. To which plants does it cause? To the rice plants. Actually, rice plants should not grow very tall because rice inflorescence is very heavy, and when it bends down in the water, then the rice seeds are getting damaged, right? So if they over soak, then they get perished, no one will buy it. So if the rice plants are going tall, then those rice plants are affected with this disease called foolish seedling disease, BOTANY disease. Then they observed, when will the rice plant get this BOTANY disease? Means if the rice plant is affected with a fungus. When affected with a fungus? When infected with fungus. You know what is the name of the fungus? The name of the fungus is Gibberella fusicori. What is the name of the fungus? The name of the fungus is Gibberella The name of the fungus is Gibberella fusicori. Gibberellins. And the earlier name of this Gibberella fusaricorum is, this fungus is Fusarium moniliforme. There is another name for this fungus which causes BOTANY disease in the rice plantlings. It is called Gibberella fusaricorum, or it is called as Fusarium moniliforme. It is also called Fusarium moniliforme, which causes the disease. So then a scientist called E. Kurosawa, he observed. What is his observation on these plants? What is the name of the scientist? E. Kurosawa. So what observation he did? Kurosawa, W-A. E. Kurosawa, he observed that now from an infected rice plant. So this is an infected rice plant which is growing very tall. So it has BOTANY disease. From the infected rice plant, he has taken the sterile filtrate. He has taken the sterile filtrate, means he collected the fluid and he gave it to the normal uninfected plant. He gave it to the normal uninfected plant. Now it is infected, means fungus will be there. When you take that filtrate and give it to the plant, this plant will show the symptoms, right? That is what is his observation. When he has taken the sterile filtrates of the infected plant and gave it to the normal plant showed the symptoms. The normal plant showed the symptoms by exhibiting this foolish seedling disease. Later, it was identified as gibberellic acid. So the Kurosawa discovery made gibberellic acid to be identified. Okay, so take a screenshot. Then we'll talk about the third hormone. The third hormone is called as cytokinin, which is also a promoting hormone, right? So what is the third hormone? It is called cytokinin. Now, a scientist is there. His name is Skoog. What is the name of the scientist? Skoog. F. Skoog. And his colleagues. Now, and his colleagues, what they have done? They were doing tissue culture experiments. They have taken the internodal stems of the tobacco plant. What did they take? They have taken the internodal stems of tobacco plant and they have taken it for tissue culture experiment. Plant tissue culture experiment. They started plant tissue culture experiment taking the tobacco internode. So when you drop it in the tissue culture medium, now what happens? A white amorphous mass will be formed which is called as callus. A white undifferentiated mass of tissue will be formed in the tissue culture medium which is called as callus. Now this callus should generate into a new plant. Morphogenesis should happen. For that we need to give plant growth hormones. Now these scientists Skoog and co-workers told that along with auxin, I told you from this callus, if you need to get root, for the callus, if the root development should happen, you have to give auxin. So along with auxin, they told you have to supplement another thing from the vascular tissue or from the DNA or from the yeast extract or from the coconut milk. When you give, then that substance which is present in coconut milk yeast extract is promoting shoot growth, they told. So when you give it, it will promote shoot growth. So along with auxin, if the medium, or they told the medium should be given along with auxin, medium should contain, they told medium should contain another substance. What is that substance? Where is that substance present? It is either present in coconut milk, we can tell, or we can tell it's there in vascular extract, or we can tell it is in yeast extract also, or we can tell it is there in DNA also. So it is there in all these things. When you give, then only what happens? Cytokinesis will happen, cell division will happen, shoot development will happen. So then it promotes cytokinesis. Cytokinesis means division of cytoplasm. Then this substance, the active substance which is present in coconut milk, DNA, yeast extract, vascular tissue, which is promoting cytokinesis, is called as cytokinin, is called as kinetin. So this substance which is present in all these things were later identified as kinetin by Skoog and Miller. Skoog and Miller named it as kinetin, which will promote cytokinesis. Along with auxin, only when you give cytokinin, then only plant growth will happen in the tissue culture medium. So this was the observation related to cytokinin. Take a screenshot, then we'll talk about the fourth hormone, inhibiting hormone, abscisic acid. Now, for abscisic acid, they did not mention the names of the scientists, but then they told 3 independent researchers, 3 scientists. What they have done, they reported the chemical characterization of 3 different hormones. Each person named it in a different manner, and they told they have inhibiting activities. Let us write down the 4th hormone is abscisic acid. The 4th hormone is abscisic acid. Now here they didn't give the scientist name. They just told 3 independent researchers. 3 independent researchers. They purified and they characterized. They have done the chemical characterization of 3 compounds. What are the names they have given? One person called it as inhibin. Okay, so what are the 3 names they have given? One person called it as abscission 2, other one called it as inhibitor B, other one called it as dormin. It's not inhibin, it is inhibitor B. One name, it is called as inhibitor B. Next name, abscission 2. Another scientist gave the name abscission 2. Another name is dormin. These are the 3 names they have given. These are the 3 names they have given, thinking that there are 3 different compounds. But later when we see, these 3 chemical compounds are the same in chemical structure, in functions also, and then all these 3 chemicals together is called as abscisic acid. Later, all 3 were proved to be the same. Proved to be the same. And then they gave the name as abscisic acid. So this is our 4th hormone. Let's go with the 5th hormone. What is that 5th hormone? Ethylene. The 5th hormone is ethylene. A scientist, his name itself is Cousins. Now, Cousins, what did he observe? He observed that when a ripened orange is kept between raw bananas, from this ripened orange, a volatile gas is released which will ripen the raw bananas also, and he named it as ethylene. So what did Cousin observe? Cousin confirmed the release of, he confirmed the release of ethylene. From where did he release the ethylene? Ethylene from ripened oranges. This ripened oranges released ethylene, and they converted raw bananas, which converted the raw bananas to ripened bananas. It converted the raw bananas to ripened bananas. So this is about the discoveries of all the 5 plant growth regulators: auxins, gibberellins, cytokinins, abscisic acid, and ethylene. Auxin by Charles Darwin and Went. Gibberellin by Kurosawa. Cytokinin by Miller and Skoog. Now for abscisic acid, no names of the scientists. For ethylene, it is Cousins. Take a screenshot, then we will talk about the physiological effects of plant growth regulators. So we have to see the functions of each hormone in detail. We will start with the first hormone, auxin. Take a screenshot, we will continue. Now let's discuss about the functions of the 5 plant growth hormones, under which the first hormone comes as auxin. Now if you recollect the information like who discovered auxin, who isolated auxin, means we told father and son, Charles Darwin and Francis Darwin. These are the people who worked with canary glass, and they identified that auxin helps in bending of the coleoptile, right? And then Wendt is a person who isolated auxin from Avena, oat plant, right? And what is the meaning of auxin? Auxin in Greek, it means to grow. That means auxin comes under plant growth promoting hormone because it helps the plant to grow, right? Then auxin was first isolated from human urine. Auxin was the first plant hormone to be identified. And where did they identify this hormone? In plant urine they identified auxin. And auxin is a normal name we are telling. If we have to see the chemical name of auxin, we call it as IAA, indole 3-acetic acid. Okay, so auxin chemically refers to indole 3-acetic acid. There are two types of auxins. The auxins which are synthesized by the plant are called natural auxins. The auxins which we manufacture them are called synthetic auxins. So there are two types of auxins, natural auxins and synthetic auxins. Examples for natural auxins are IAA and IBA. Example for synthetic auxins are NAA and 2,4-D. So many a times in NEET examinations they have asked IAA is example of naturala, synthetica, NAA is example of naturala, synthetica. So IAA and IBA comes under natural auxin. IAA stands for indole acetic acid. IBA stands for indole butyric acid. Coming to synthetic auxins, NAA stands for naphthalene acetic acid and 2,4-D stands for 2,4-dichlorophenoxyacetic acid. These two are synthetic auxins, right? And when it comes to the functions of auxin, now what are the roles that auxin has with the plant? That if we have to discuss, now auxin initiate roots in the stem cuttings. Now if we go to nursery and if we buy a small plant, they'll give us it in black colored cover. Now this much tiny plant, it will be there. When you open that black colored cover to keep it inside the pot, if you see the root system for that tiny plantlet, profuse of roots will be there. Means how did the gardener generate those many roots for this much small stem cutting? Means they will cut the matured stems, they'll dip it in auxin solution. Auxin promotes rhizogenesis. Auxin promotes root development. When you dip it in dilute auxin solution, roots get generated. So that's how the gardeners will propagate the plants. If you plant the seed, that seed has to grow and it has to come in. It will take too much of time. But vegetative cutting, if you do, you can plant, you can generate so many plants by dipping the stem cuttings in auxin. That is the first function. In pineapple, what do we eat is fruit. Now if there are more flowers, then more fruits, and the farmer will get more money. So it promotes flowering in pineapple. Auxin will promote flowering in pineapple. Second function, for stem cutting, it initiates roots. First function, now it prevents early fruit fall and leaf fall. Leaf is a food factory. The more the leaves, the plant will do more photosynthesis, it can manufacture more food. And then if the fruits, early fruits, if they fall off, then they are wasted. Harvest will be less. So auxin, it prevents leaf fall. Good. It prevents early fruit fall also, but it promotes senescence of the older fruits and older leaves. Older leaf has to fall down, right? It cannot do photosynthesis. Older fruit, when it gets ripened, that also should fall down. So it promotes senescence in the older parts, but it prevents early fruit fall and leaf fall. Third function, good function. Next one, it causes apical dominance. What do you mean by apical dominance? Auxin, usually it's present at the root tip and the shoot tip. Now this auxin which is present at the tip, that will make the plant to grow tall. It will make the plant to grow tall, and when the apical bud is growing, this auxin which is present in the apical bud will have an inhibitory effect on the lateral buds. Lateral buds cannot grow. So that means the plant will not become bushy, the plant will grow very tall. So that concept of inhibiting the growth of the lateral buds by the apical bud is called apical dominance. Auxin is responsible for apical dominance. Auxin causes apical dominance, means the apical bud inhibits the growth of the lateral buds, means the plant will not become bushy. Now if we cut down the apical bud where auxin is there, I told you auxin is synthesized at the shoot tip and the root tip. Cut down the shoot tip, that's called decapitation. If we do decapitation, there's no auxin, we removed it off, then the dominating factor is not there, then aram se these lateral buds will grow and the plant will become bushy. If we see tea plantation, the people will be plucking the tea leaves at their height, means it's a bushy plant, it's not growing tall. We need the leaves, they used to pluck and they'll drop it, they'll pluck and they'll drop it. So for tea plantation and for hedge making, bushy making, so auxin should not be there, means we have to do decapitation. Decapitation help in hedge making and tea plantation. It's not tree plantation, it is tea plantation. Right. Next one. Auxin induces parthenocarpy in tomatoes. What is parthenocarpy? The fruit which is formed without fertilization is called parthenocarpy. Auxin promotes parthenocarpy in tomatoes. Next one. Excuse me. 2,4-D is an example of synthetic auxin. 2,4-D acts as a herbicide. It acts as a herbicide, it acts as a weedicide. Now when we make lawn, in that lawn only grass should grow, which is a monocot. But if any dicot is growing as a weed, now it will become visible and it will look odd. So dicot weed should not grow. If monocot grasses are growing, then no issue in that grass, in the lawn grass, this grass also will mix up and it's not that much prominent to get identified. Whereas dicots in a lawn easily get identified. So that's why when we spray 2,4-D, a synthetic auxin on the lawn grass, what happens? No effect on the lawn grass. Monocots, it will not show effect, but it will kill dicot weeds. So hence it is used to prepare weed free lawns. It kills the dicot weeds. Who? 2,4-D. Next one. Auxin controls xylem differentiation. Auxin will help in root development. Function of the root is to absorb more water and transport it to all the parts of the plant. Now who will help in transport of water? Xylem. That means auxin helps in xylem differentiation also, so that the root will have more number of xylem elements, tracheids and vessels, and it can help in better conduction. And auxin is initiating root growth, no? So that means auxin helps in cell division also. If we have to summarize the functions of auxin from the last, cell division it will do. Then next one, xylem differentiation it will do. Weed free lawns it will maintain. Then parthenocarpy in tomatoes. It will cause apical dominance. When you cut down the apical bud, lateral bud will develop and hedge making can be done, tea plantation can be done. It induces flowering in pineapples. It will not allow the early leaves and early fruits to fall, but older leaves and older fruits will fall down. And auxin initiates root in the stem cuttings. So take a screenshot. We finished auxin. Then we'll talk about the second hormone, that is gibberellin. Right. Now what is the second hormone? The second plant growth regulator is gibberellin. And if you remember, now gibberellin is named after the fungus called Gibberella fusicori. Yeah, these points I forgot to tell. Auxin is synthesized from an amino acid called tryptophan. Auxin is synthesized from an amino acid called tryptophan, and zinc is required for auxin synthesis. This point we studied in mineral nutrition. Zinc is required for auxin synthesis. And auxin is synthesized from which amino acid? Because indole compound, no, tryptophan also has indole structure in it. Now let us talk about the second hormone. What is that? It is gibberellic acid. Now, who is that scientist who isolated gibberellins? Means he is a Japanese plant pathologist. His name is E. Kurosawa. We studied this in discoveries, right? So he is a person who observed that the rice plants were infected with a fungus called Gibberella fusicori. They grow very tall, foolish seedling disease, BOTANY disease, we will tell. So that was his discovery. But then who crystallized gibberellin? Who took the gibberellin out? Who gave the chemical nature of gibberellin out? Means Yabuta and Sukumi. So if we tell Japanese plant pathologist. Okay, what is his name? Kurosawa. Now he identified that the rice plants which are infected, the rice plants which are infected with fungus. What is the name of the fungus? Gibberella fusicori. Right? So if the rice plants are infected with Gibberella fusicori, then they develop foolish seedling disease or BOTANY disease. Then it shows what disease? BOTANY disease. What do you mean by BOTANY disease? BOTANY disease is also called foolish seedling disease, right? Now, but who crystallized gibberellin? So, gibberellic acid was crystallized by, Iebuta, another Japanese scientist, Sukumi. These are the people who crystallized gibberellic acid. Now, so far, how many gibberellins have been identified? Till now, 125 gibberellins are identified. But the first isolated gibberellin, the most extensively studied gibberellin is gibberellic acid 3. So, we can tell till now 125 gibberellic acids have been identified. They have been identified, but then out of this, which is the first gibberellic acid to be identified? So the first gibberellic acid is, it is called GA3. All those gibberellic acids, GA1, GA2, GA10, GA125, like that. So the first gibberellic acid to be identified is GA3, and it's the most extensively studied gibberellic acid also, and it is the most extensively studied hormone. Right? Now, gibberellic acid is acidic or basic. The name itself tells it is acidic. And gibberellic acid is derived from which chemical? Gibberellic acid is derived from terpene compounds, right? And is a promoter inhibitor. It is a promoter only. Now, gibberellic acid physiological functions, if we have to list down one by one, let us see. The physiological roles of gibberellic acid are: the first function we can tell. Now, if we buy a grape stock, in that the grapes are clustered together, they are congested together because the stalk of the grape is small and grapes are big. So because of that overcrowding, what happens? They get crushed when they are packed up. Now imagine if the stalk of the grape is long, then one grape is separated from other grape by a distance, then it will be appealing also, and wastage will not be there. So if we spray gibberellic acid, then what happens? It increases the length of the stem axis in the grape stalk. So the first function, it increases the length of the stem axis of grape stalks, so that each grape is separated from each other nicely. There will not be any crushing and they will not spoil easily. Another thing, now the second thing is, it elongates the apple size. It improves the shape of the apple. Now if you spray gibberellic acid on apple plantation, what happens? Apples will elongate in their size, and there will be improvement in the shape of the apple also. So gibberellic acid elongates apple, and also it improves the shape of the apple. The shape of the apple also will be good. This is the second function of gibberellic acid. First function with the grape, second function with the apple. Third function we can tell, like auxin also, what it will do? It will do senescence of the older parts. Here, this thing also, it delays senescence. There it will promote senescence of the older parts. This will delay senescence. Old age is called senescence. It delays senescence. Since it delays senescence, the fruits are left on the tree for the longer time. Since it delays senescence, we can tell the fruits are left on tree for longer time. So if all the fruits ripen at the same time, you'll cut it, you'll take it to the market. In the market, if so many are there, then there's no demand for that, then the cost becomes low. If you cut 10 kgs one day, 10 kgs one day, that demand will be created, and you can negotiate on the cost. You can take it to a high cost. So it increases the market life. Okay. So by delaying senescence, by keeping the fruits on the tree for longer time, the market period is increased. The market period of the fruits is increased. This gibberellic acid will do. Next function, if we have to talk about, now brewing industry means alcohol industry. For that, the starting material is barley or rice or fruit juice, like that. Now if we spray gibberellic acid to that brewing industry, it speeds up the malting process of the barley. The barley undergoes malting process very fast, so alcohol production will be done very fast. Speeds up the malting process in the barley during alcohol industry, beverage industry. It speeds up the brewing, it speeds up the malting process. Where? Malting process in barley. In which industry? Alcohol industry, which is called as brewing industry. So if the malt formation happens fast, then the alcohol conversion also happens first. Then next one, it will increase the internodal elongation, right? It will increase, it's increasing the stem axis. In the sugarcane, what we take is the stems only. Now in the sugarcane plantation, if we spray gibberellic acid, then the stems will grow even long, and we get more amount of sugarcane, we get more money. Per acre, you can increase the harvest by 20 tons. So if we spray gibberellic acid in sugarcane plantation, now what will it do? It will increase the stem. It increases the internodes. It increases internodes, and it will give us more harvest. How much harvest? 20 tons per acre it will increase. 20 tons per acre. 20 tons per acre it will increase. So this becomes the next function. Now if we talk about rosette plants, cabbage, cauliflower, lettuce, beetroot, all these are rosette plants. In rosette plants, what it will do? It promotes bolting. What do you mean by bolting? It promotes bolting in rosette plants when just before flowering, so that the flowers and all will be above the soil surface. If cabbage, cauliflower, if they are on the soil surface, insects and all will infect it easily, and we don't eat it. So now if it promotes bolting, then it will grow on the top, right? So it promotes bolting in which type of plants? In rosette plants, when just before flowering. That's the main function of gibberellic acid. It promotes bolting. Gibberellic acid promotes bolting in which plants? In rosette plants. Promotes bolting in rosette plants. Examples of rosette plants: cabbage, we can take cauliflower, we can take beetroot, we can take. Now all these are rosette plants. It promotes bolting in rosette plants like cabbage, cauliflower and beetroot when just before flowering. So it promotes bolting just before flowering. So what did we discuss, children, so far related to gibberellin? So we told gibberellic acid was first identified by a Japanese scientist called E. Kurosawa. So he told that rice seedlings will get infected if they get infected with a fungus called Gibberella fusicori or Fusarium monelifame. They get a disease, foolish seedling disease called BOTANY disease. But who crystallized it? Yabuta and Sukumi. They crystallized gibberellic acid. How many gibberellic acids are so far identified? More than 125, but which is the most commonly studied one? Ga3. It is acidic. Coming to the roles, it will increase the length of the grapes. It will increase the shape of the apple. Next, it delays senescence so that the fruits will retain on the tree for more time. Next one, it speeds up the malting process. So brewing industry, it'll do very fast. Next one, it increases the sugarcane yield 20 tons per acre, and it promotes bolting in rosette plants like cabbage, beetroot, sugarcane and all when just before flowering. Take a screenshot. Two other functions are there. We'll talk about that also. Alright, so 7th and 8th functions also. Let us talk about gibberellins. Conifers means cone shaped trees. Actually conifers will take long time to get matured. So when the plant is in the juvenile stage, then itself if you spray gibberellin, it grows fast, it gets matured fast, it will undergo fertilization fast, and it will produce seeds fast. So 7th function we can tell, spraying gibberellin in juvenile conifers will fasten the maturity period. So 7th function is spraying gibberellic acid on what plants? On juvenile conifers. What it will do? It will fasten the maturity period. It matures faster. It will fasten the maturity period. If it matures fast, then the seed production also happens fast. So leading to seed production early seed production will happen. This is the next function of gibberellic acid, right? Then if we have to talk about the bioassay test, for Auxin bioassay test is Avena curvature test. Now let us talk about the bioassay test for gibberellin also. What is the bioassay test for gibberellin? There are two tests for gibberellin bioassay test. The first test is called lettuce hypocotyl elongation. Lettuce, cabbage, cauliflower, the stem is elongating, right? It is bolting. So lettuce hypocotyl elongation test is one test for identifying gibberellin. Let me write on. Lettuce hypocotyl elongation test. And another test is brewing of barley, right? That means barley means it is cereal. In cereal, endosperm, which is a nutritive tissue, if it digests fast, then the brewing will happen fast. So the second bioassay test is cereal endosperm digestion test. The second test bioassay test is cereal. Nutritive tissue is endosperm digestion test. These two are the bioassay tests for gibberellins. Avena curvature test is bioassay test for Auxin. Take a screenshot. We finished gibberellin. Then we'll talk about the third hormone, cytokinin, which also comes under plant growth promoter only. Now, related to cytokines, if you see, Skoog and Miller are the scientists who isolated cytokinins, and they told it has cytokinesis effect. It will promote cytoplasm division. So that's how it got the name called cytokinin. Now let us talk about the third plant growth regulator, Amma. What is that? Cytokinin. Now who isolated it? Skoog and Miller. They named it as kinetin. They named it as kinetin. What it will do? It promotes cytokinesis. So this kinetin, it got its name because it promotes cytokinesis. Cytoplasm division is called cytokinesis, right? Now, from where did they isolate cytokinin? Auxin is isolated from urine. Gibberellic acid is isolated from where is rice plants which are infected with Gibberella fusicoria. Cytokinin is isolated from a fish called herring fish. It has sperm. From autoclaved herring sperm DNA, they isolated cytokinin, or they isolated kinetin. So we can tell kinetin was first isolated from where? From autoclaved herring, which is a fish, from its sperm DNA. They isolated kinetin, and they thought naturally kinetins are not there. So they were in search of kinetin like hormones. So they were in search of kinetin like. What kinetin will do? Cytokinesis. Cytokinesis promoting hormones. They were in search. Then they found a hormone called zeatin. which will also do cytokinin. From where did they find the zeatin? From maize kernel and coconut milk. Zeatin, which also has the same roles of cytokinin or kinetin, was isolated from coconut milk, and it was isolated from maize, sweet corn, right, kernel. From here they isolated. From then onwards many cytokinins have been identified, right? Now where is cytokinin synthesized? Where is cytokinins present in more? Cytokinin should be present in those regions which are actively dividing, because cytokinesis means active division, means at the root tips, shoot tips, leaf, flower, right? Floral buds. These are the regions where cytokinin will be more. So we can tell that cytokinins are synthesized more. Where are they synthesized more? In apical regions, root apical region, shoot apical region, and in the buds where active division occurs. Wherever cell division is more, there and all cytokinesis is required. There cytokinin is synthesized more. Coming to the physiological functions of cytokinin. Coming to the physiological functions of cytokinin. Cytokinin will promote more leaf formation. So more leaf formation, it will promote more leaves. First function. Next function, if we have to write, cytokinin, it promotes more chlorophyll synthesis also. So more chlorophyll synthesis also it will do. So leaf synthesis it will do more, chlorophyll synthesis also it will do more, and then lateral shoot development. Lateral shoots also will be grown more. Lateral shoots are stopped by auxin. Lateral shoot growth it is promoting, means cytokinin is having opposite action to auxin with respect to apical dominance. So cytokinin promotes lateral shoot growth. That means it is having opposite role to auxin. In which concept? In the concept of apical dominance. In the concept of apical dominance, auxin is having opposite role to cytokinin. Not only lateral shoot formation, cytokinin also has role in adventitious shoot formation. If the shoot is formed from the plumule, it is normal shoot. If the shoot is formed from any other part other than the plumule, it's called adventitious shoot formation. Cytokinin, it also helps in adventitious shoot formation. Cytokinin helps in adventitious shoot formation. It overcomes apical dominance, and cytokinin prevents leaf senescence. It helps the leaves to grow, right? So it will not allow the leaves to fall. It promotes, we can tell it delays leaf senescence. Last function, it will delay leaf senescence. It will delay leaf senescence so that whatever the nutrients are there on the leaf will be mobilized off. That means it is promoting nutrient mobilization by delaying the leaf senescence. It delays leaf senescence, and what it is doing? It promotes nutrient mobilization. So these are the 5 functions of cytokinins. Now let's take a recap. Cytokinin was discovered by Skoog and Miller. They named it as kinetin. Why? Because it has role in cytokinesis. It was first isolated from herring sperm fish DNA. They thought natural kinetin is not there. When they went in search, they found a kinetin-like compound which is called zeatin from coconut milk and maize kernel. Right? Cytokinin, since it is promoting cytokinesis, now wherever the apical tips, root tip, shoot tip, leaf buds are there, where cell division is happening, their cytokinin is synthesized in more concentrations. Coming to the first function, more leaf formation, more chlorophyll formation. Then we can tell lateral shoot formation, adventitious shoot formation, prevents apical dominance, delays leaf senescence, and promotes nutrient mobilization. So with this we completed auxin, gibberellin, cytokinin. Take a screenshot. Then we will talk about the next hormone, inhibiting hormone called abscisic acid. right? Now what is the fourth hormone? It is abscisic acid. Now 1, 2, 3, these are promoting hormones, but abscisic acid is an inhibitor hormone. And if we recollect who discovered abscisic acid, they did not give us the scientist names, but they told 3 scientists independently. They identified one person, named it as inhibitor 2, abscission 2, inhibitor B, and dormin. Later they identified all these 3 substances are the same, and they called it as abscisic acid. Okay, now let us talk about the next hormone, abscisic acid. Right, so this is also acidic substance. So this is also acidic hormone. It is not a promoting hormone, it is a plant growth inhibitor. So abscisic acid acts as plant growth inhibitor. Abscisic acid acts as plant growth inhibitor. Now what it will do? So it will do seed, it will delay senescence. What it will do? It inhibits seed germination, it induces dormancy. So abscisic acid induces seed dormancy, means it will not allow the seed to germinate. It will not allow the seed to germinate. It is asking the seed to undergo dormancy. It induces seed dormancy, means it is inhibiting seed germination. So we can tell, in turn, abscisic acid, it inhibits seed germination. If it is doing dormancy, means then it will not allow it to germinate. Now abscisic acid, they help in closing the stomata. They help in closure of stomata. When it will help in closure of stomata? During stressful conditions. It helps in closure of stomata during stress. So that is why abscisic acid is also called stress hormone. It is also called stress hormone. Why is abscisic acid called stress hormone? Means it will allow the plant to combat the stress, okay, to tolerate the stress. So when the plant is wounded, so first it has to heal itself. It should not do growth, it should not undergo development. First it has to heal itself. So that's why it will allow the plant to overcome the stress. In that conditions, what it will do, it will not take carbon dioxide and it will not do photosynthesis. So for that it has to close the stomata, right? But abscisic acid, important role in seed development. It is asking it to complete its dormancy, then it helps in seed development. It helps in seed development, it helps in seed maturation, and it helps in seed dormancy also. So abscisic acid has important roles with seed development. No, with seed development, we can tell with seed dormancy and with seed germination. So after completing the dormancy only, it will allow the seed to germinate. So it has role in, that means it has role in seed maturity. Abscisic acid has role in seed maturity, and most of the functions of abscisic acid are opposite, are antagonistic to gibberellins. This they asked in NEET examination. Most of the roles of abscisic acid are antagonist to gibberellic acid. Gibberellic acid is a promoting hormone. Whatever it will promote, abscisic acid is inhibit. So if that tells yes, this will tell no. If that tells yes, this will tell no. So both of them have opposite actions. Now let us summarize abscisic acid. We don't know who discovered abscisic acid. They just told 3 researchers independently discovered and they named them as 3 compounds. But when we see the chemical natures, they are the same. That's why we call it as, together we call it as abscisic acid. It is an acidic substance. It is derived from carotenoid compounds. And this abscisic acid, it is a growth inhibitor. So it will cause dormancy, it will inhibit germination. Now it will close the stomata, it will allow the plant to manage the stress. So that's why it's called stress hormone. It has very important role with seed maturation, seed development and seed dormancy. The last important function of abscisic acid is all the functions of abscisic acid are opposite to gibberellic acid. Take a screenshot. We'll talk about the next and the last hormone, which is ethylene. We'll discuss about the last hormone, which is ethylene. So there are so many roles of ethylene. You remember who isolated ethylene? Ethylene was identified by Cousins. Cousin is the name of the scientist who isolated ethylene. From where? From ripened oranges. It's a simple hormone, it's a volatile hormone, it's a gaseous hormone, right? Now, what are the roles of the ethylene? Ethylene helps ethylene is synthesized in more concentrations in which tissues? In the tissues which are undergoing senescence, which are undergoing ripening. Ethylene is synthesized in more concentrations in the older tissues and the ripened fruits. First function, ethylene promotes horizontal growth of the seedling. Even if the seed is kept horizontal, then also if ethylene is there, it can generate root down and shoot up. And swelling of the axis and apical hook formation in dicots. Only in dicots, in between the cotyledons, apical hook will come, from there the pumil will come. Ethylene helps in apical hook formation in dicots, horizontal growth of the seedling. Third function, we wrote here, it will promote senescence. So it promotes senescence, old age. It promotes abscission. It will allow the older leaves to fall, older fruits to fall, and it is a fruit ripening hormone. Ethylene basically is a fruit ripening hormone. At the time of ripening, it increases the respiration enormously, which is called as respiratory climacteric. So just before ripening, so the ethylene will increases the respiratory rate of the fruits, which is called as respiratory climacteric. And ethylene will break the bud dormancy, seed dormancy. Abscisic acid will cause dormancy. This will break dormancy. And since it is breaking seed dormancy, it will allow the peanut seeds to germinate. It will allow the potato tubers to sprout and generate their plants. It promotes rapid internodal elongation in the deep water rice plants. If the internodes elongate more, then the plants will not be very dwarf, they'll be little tall, so that the rice plants will not dip inside the water. It is good only. Then it promotes root growth. It promotes, auxin will promote root growth, ethylene also will promote root growth. Root hair formation also it will increase, so that the surface area for absorption of more water and more minerals increases. Then it initiates flowering. It will initiate flowering and it will make all the flowers to ripen, the fruits to ripen at the same time. It's called synchronization, so that the farmer can cut all the pineapples at a time, so he can save his labor cost. So that's called synchronization of the fruit set. Now it initiates flowering and synchronization of the fruit set. Then it induces flowering in mango, so that if flowers are there, fruits will come. Then most widely used plant growth hormone in agriculture. So many functions are there. Why we will not use? We will use it. Then liquid form, since it's a gaseous hormone, if you spray it in the gas, it goes to the atmosphere, it gets wasted off. So that's why the liquid form of ethylene is called ethafone. It will be slowly released because it is a liquid state. Ethafone will fasten the ripening. Ethylene is a ripening hormone. Ethafone also will be a ripening hormone. It will ripen tomatoes, it will ripen apple. Now we know it accelerates senescence, it accelerates abscission, so it will accelerate abscission in flowers also, fruits also, leaves also. It will thin the cotton. If the cotton fibers are thin, then it will be of lightweight, so it will be appealing. It will thin the cherry wall also. It will thin the walnut wall also. And if we take cucumber is dioecious, right? Cucumber is means monoecious. Male and female flowers will be there on the same plant. Now it converts the male flowers into female flowers, so that they convert into fruits, so we get more yield or more harvest. So these are the functions of the ethylene. So basically what it is, it will promote ripening. So it's a ripening hormone, fruit ripening hormone. Cousins observed it, right? And it causes respiratory climacteric, and it breaks bud dormancy, seed dormancy. It allows the peanut seeds to germinate. It allows the potato tubers to sprout, and it promotes internodal elongation of the rice plants. Then it promotes root growth. It promotes root hair formation, so that it increases the absorption rate. Then it initiates flowering and synchronization of the fruit, so that the farmer can cut them at the same time. It makes the mango to flower more, so that the harvest harvest will be more. And the liquid form of ethylene is called ethaphon, so that it will not get wasted, it will be released slowly. It is used for ripening vegetables also, it is used for ripening fruits also. It will thin the cherry, it will thin the walnut, and it will thin the cotton. And it converts the male cucumber flowers also into female cucumber flowers, it increases the yield. So we finished all the 5 plant growth hormones. The last 2 topics are photoperiodism and vernalization. Take a screenshot. We will continue with photoperiodism. So the next topic is photoperiodism. So we completed all the plant growth hormones, auxins, cytokinins, gibberellins, abscisic acid and ethylene. So the last 2 topics which are pending in the chapter are photoperiodism and vernalization. Let us see that also fastly so that we can finish off the chapter. Now the name itself tells what do you mean by photoperiodism. So there are some plants, now they require light treatment before flowering. Okay, they will flower only when they are exposed to that particular period of light what they require, right? Not only that, they measure the light also, how many hours they are getting exposed to the light, they measure also. Now this phenomenon is called as photoperiodism. Now how much amount of light, there is a period which is called as critical duration. We can call it as critical photoperiod. Now the critical photoperiod from plant to plant, it changes, it varies. That is what they told in NCERT plan. Now if we take the critical photoperiod as 10 hours or 11 hours, let us take 11 hours. Now the plants which require more amount of light than the critical photoperiod, right, means the plants which are requiring more than 11 hours are called as long day plants, right? So let us see here, we wrote here photoperiodism. Who explained the concept of photoperiodism? Garner and Allard. In the year 1920, they explained the concept of photoperiodism, right? In which plant they observed photoperiodism? They observed photoperiodism in Maryland mammoth, which is a mutant variety of tobacco. So they did experiments in a mutant variety of tobacco which is called Maryland mammoth. Who are the scientists? Garner and Allard in the year 1920. So they are telling that some plants require a periodic exposure of light. Some plants require light to induce flowering so that they can flower, right? Now these plants, they even measure how many hours of light they are getting exposed to, even they can measure that, right? Now I told you there is a concept called critical photoperiod, right? Now those plants that require exposure of light more than the critical period. I told the critical photoperiod keeps changing from plant to plant. It can take it as 10 hours, you can take it as 11 hours, we can take it as 12 hours. Now if you take the critical photoperiod, now the plants which are requiring light more than the critical photoperiod are to be called as long day plants. So they are called as long day plants. On the other hand, the plants which will flower when you expose them to light which is lesser than the critical photoperiod, which is below the critical photoperiod, are called as short day plants. The other set of plants will be there, so they are not stimulated when you expose them to light. So they finish off their vegetative phase and at maturity they flower by themselves. They don't require light induction to flower. So then light is not showing any influence on them. So call them as day neutral plants, right? So long day plants means they require light exposure more than the critical period. Short day plants means the other plants, they require light exposure lesser than the critical period. They are called short day plants. Now I told the third category where there are many plants where there is no correlation between the light exposure and the flowering. So they are not induced when they are exposed to the light. So then such type of plants which flower all throughout the year are called as day neutral plants. They are called as day neutral plants. This is a picture which is there in NCERT. We need to understand these pictures also. Now they told this category of plant is called long day plant, this category of plant is called short day plant, and this category of plant is called day neutral plant. That's what they showed in NCERT. And then they marked a line, and this line they told it is critical photoperiod. This line is critical photoperiod. Now in long day plants, they require light above the critical photoperiod, no? So that's what we write. They require light more than the critical photoperiod. Now this is the critical photoperiod. Above the critical photoperiod, above the critical photoperiod, if you supply light, then they flowered. Below the critical period you supplied, they did not flower. Now come to short day plants. Short day plants will flower when they are exposed to light lesser than the critical photoperiod. They will flower. Now see, this is below line and this is above line. This is a critical photoperiod. Now if they are exposed to the light below the critical photoperiod, the bottom side is below, the upper side is above. So if they are exposed to the light below the critical photoperiod, they flowered, then they are called short day plants. Whereas day neutral plants, they don't have a concept of critical photoperiod only, just they will complete their vegetative phase, they'll reach their maturity and they flower. That's what they told. They flower after their vegetative maturity is achieved. When they complete their vegetative period, they start flowering. Then what to call them as? Day neutral plants. Of course in NCERT they did not give us the examples for short day, long day and day neutral, but if the paper is going to be tough, they will ask us the examples. So why to take a chance? Let us remember some examples for long day, short day and day neutral plants. Under long day plants, the examples are, write down the examples and just remember them. Under the long day plants, the examples are, one is spinach, other one is spring barley, third one is beetroot, sugar beet, henbane, wheat, radish, oat, lettuce and larkspur. Usually we'll make a code to remember the examples, but here so many plants are there, so I felt like making code is difficult, so we will remember normally itself. So long day plants, if you see, right, spinach, spring barley, sugar beet, henbane, wheat, radish, oat, lettuce, larkspur, these are the examples of long day plants. And coming to short day plants, now it was worked by Garner and Allard on Maryland Mammoth mutant variety of tobacco, which is a short day plant. So that is the first example. Maryland Mammoth variety of tobacco is the first example. Xanthium, chrysanthemum will come in the winter season only, where the day duration is less, right? So xanthium, rice also, chrysanthemum, all these are flowering plants. Then tobacco, yeah, we know. Potato also, aloo also will come in the winter season only. Then soya bean, dahlia is also the beautiful flower which is used in decorations and all, which is used in bouquet makings and all. That also is coming winter only. Aster flowers also. Then bryophyllum, which is an example for reproductive leaf, that is also short day plant. Then strawberry also. So you can remember most of them are in the winter only. In the winter time only, the day length will be lesser than the night duration, right? So the short day plants, I think we can remember. Tobacco is one example. Then we can take the example of xanthium, chrysanthemum, strawberry or potato, dahlia, aster, then tobacco. All these are the rice, all these are the examples for short day plants. Coming to day neutral plants, the examples of day neutral plants are cotton, cucumber, maize and tomato. Cotton, cucumber, maize and tomato. So remember these examples for long day, short day and day neutral plants. Now when we come to the concept, in the recent years they came to know that it's not only the day duration they are measuring, they measure the night duration also. Means the amount of daylight what they require and the amount of night light also they are counting. That's what they wrote in NCERT. It is not only the duration of the light duration, they measure the duration of the dark also. Darkness is also equally important for them. That dark period, if you are interrupting it by exposing it to a flash of light, then dark period is interrupted, then the short day plant will not flower. Okay, so not only the duration of the light, the duration of dark is also equally important. This response of plant to the periods of darkness also, to the periods of light also. So we can write light and night. We can write light and night. It's called photoperiodism. So to how much duration of light the plant should be exposed, or to how much duration of night the plant should be exposed so that it can induce flowering. This concept, this phenomenon is called as photoperiodism. Now we are talking about light. That means a light is a solar source, right? Now light is absorbed by a pigment. Now where are pigments present? On the root or stemma, leaf. Now we know that chlorophyll is a pigment and other carotenoids, xanthophylls, all these are the pigments. Where are they present? They are present on the leaf. That means who will perceive the light? Who will perceive the light? Leaf will perceive the light. Now when the leaf contains pigments, when the leaf is exposed to light, when the leaf traps the light, then in this leaf what happens? The pigment of phytochrome gets activated. That phytochrome will synthesize a hormone, which is a hypothetical hormone. Why I am calling it as a hypothetical hormone means it is not even isolated. Means scientists strongly believe that the hormone is there, but they were not able to isolate it. Now the leaf will synthesize a hormone called florigen, and this florigen transports from the leaf to the shoot. Why it is transporting from the leaf to the shoot? Because the shoot tip only transforms into a flower. It transforms into floral primordium. So the florigen is responsible for flowering, and the florigen is synthesized by the leaf when the leaf is exposed to the required duration, right? That's what they wrote here. It is also interesting to note that the stem is a modified flower. Stem is modified into flower, but who is perceiving the light? Light is perceived by the leaf. When the leaf is perceiving the light, when the leaf is receiving the light, then it will synthesize a hormone. What's the name of the hormone? Florigen. This florigen, which is synthesized at the leaf, migrates to the stem. Then the stem gets condensed and it becomes a floral primordium. From there the flowering starts. Children, this is the concept of photoperiodism. Who discovered it? In which plant they discovered it? Now what are the definitions of long day plants, short day plants and day neutral plants? Is it only the day length or night length is also equally important? Night length is also equally important. Who will perceive the light? Leaf will perceive the light. But where is flowering happening? On the stem. What is that hormone which is responsible? Florigen is a hormone responsible. Now what is critical photoperiod? Critical photoperiod is that period. Now the plants which require more than the critical photoperiod, long day. Which require less than the photocritical period, short day. Which doesn't have any photocritical period, day neutral. These are the examples. Take a screenshot, then we'll talk about the last concept of the chapter, which is called vernalization. Right, so the last concept of the chapter is vernalization. Now since you have asked for one-shot videos, we are making these videos. Now afterwards I'll post you some questions from this chapter also. So after watching this lecture, you can attempt solving those questions. So it will be helpful for you to score more. Even if your concept is clear, unless and until you solve MCQs, you don't know whether you completely understood the concept or not. So I suggest you, after reading, you have to work out the MCQs also. Now the last topic is vernalization. Right? Now, who coined the term vernalization? A Russian agriculturist or a Russian agronomist. His name is Lysenko. The term vernalization, who coined it? Was coined by Russian agronomist. What is his name? Lysenko coined the term vernalization. And who explained or who has done the experimental verification for vernalization means it is by Klippart. Experimental verification. Right, experimental verification of vernalization was conducted by which scientist? Klippart. Conducted experiments on the concept of vernalization. Now what is vernalization? There are some plants which flower either qualitatively or quantitatively. So when they will flower? When they are exposed or when they are subjected to low treatment. Okay, so plants will flower only when they are subjected to low treatments, cold treatments. Then that concept is called vernalization. Right, let us write down. There are plants for which flowering is either qualitative or quantitative. There are some plants for which flowering is either qualitative or quantitative. Now what do you mean by quantitative? The number of flowers. Qualitative, the size of the flowers. Qualitative or quantitative effect will be there on flowering when they are exposed to low temperatures. On exposure to low temperatures means induction of flowering subjecting the plant to cold treatment is called vernalization. In simple, if you have to tell, if this statement is difficult for you, you can say the other way also. So subjecting the plants to chill treatment and inducing flower is called as vernalization. Now when we talk about what is the significance of vernalization, let us talk about some varieties, right? We told some plants. What are those plants? There are some food plants like wheat, barley and rye. If we take food plants, what are the examples of food plants I am telling? One is wheat, the other one is barley, and the third one is rye. Now these plants, food plants, will have two varieties. One is called as spring variety and the other variety is called as winter variety. So there are two varieties, right? Spring variety and winter variety. Now spring variety of wheat, if you take and if you sow them in the spring season, then the seed will germinate, it will develop into seedling and it will produce, it will grow, it will reach its vegetative period. By the end of the growing season, you can harvest them. So spring season, spring variety plants, if you plant them in the spring season, now nicely they will germinate, they will develop into plantlets, and by the end of the growing season, we can harvest the grain. So there's no delay in this. But when you take the winter variety and when you plant it in the spring season, they fail to germinate or they fail to grow and they fail to give you grains. Now when do they sow this winter variety wheat? Then winter variety wheat, if it is planted in the autumn season, when you plant them in the autumn season, then the seed will germinate, then it will develop into seedling. Now after autumn season, winter season will come. In that winter season, this small seedling or the plantlet which is developing, now it will receive the amount of chill. Winter means chill season. It will receive that cold treatment. After receiving the cold treatment only, it will start flowering. Flower converts into fruit and grain. So winter variety requires vernalization. So that is why winter variety, if you are planting it in the spring season, it is not giving you the flower, fruit and all. Why? Because it needs that vernalization. So that is why spring season, if you plant in the spring variety if you plant in the spring season, they grow and the grains can be harvested. When can we harvest? The grains can be harvested at the end of the growing season. At the end of the growing season. And if you take the winter variety, if winter variety is planted in the spring season, if winter variety is planted, if it is planted in the spring season, will it germinate or it fails? It fails. Then when should we plant the winter variety? Winter variety should be planted in the autumn season then. So but if planted in autumn season, then what happens? Tell me. If you plant in autumn season, then the seedling will grow. Then in the winter season, it is subjected to that cold treatment. After taking that cold treatment, then it starts flowering and you can harvest the grains. And you can harvest the grains by midsummer. By midsummer you can harvest the grains. Means this winter variety requires that particular chill treatment. Okay. Now this is the concept of vernalization. One more example we can take for vernalization, that is biennial plants. Biennial plants means whose lifespan is 2 years. 1 year they'll try to complete their vegetative growth, means root, stem, leaf will grow. They'll reach to their maturity. In the second year they will flower and fruit and then they will die. So this is biennial plant story. Now biennial plants, now why are they waiting for that year-end calendar? Means they are waiting for that chilled treatment. Root, stem, leaf, everything got developed. They are waiting for that winter season. They are waiting for that cold exposure to flower, fruit and produce the grains and all those things. Now if we subject them to cold treatment before itself, then they will flower and they'll produce the fruits and all, and then they will die. That means biennial plants can be converted to annual plants, means they can be flowered fast, they'll produce their fruits fast when you subject them to cold treatment. This is another example. What is another example? Another example is biennials. What are biennials? All the root plants, carrot you take, radish you take, right, and beetroot, cabbage, right, all the rosette plants, they are biennial, means first year they will grow, vegetative growth will happen, in the second year reproductive growth will happen, flower, fruit formation will happen. Now why are they waiting till the end of the year? Why are they flowering in the second year only? Because they are waiting for that chill treatment. Now if you artificially give that chill treatment in the 6th month or 7th month itself, then after finishing the vegetative growth, then what happens? You have given the chill treatment, then it will flower and it will produce fruits, and then it's died, it's monocarpic, because biennial plants are monocarpic. They are monocarpic. Once they flower, once they produce the fruits and seeds, they will die. Okay, so they will die. Okay, now when we subject them to the cold treatment artificially, we can convert biennial plants to annual plants. So this is one advantage of vernalization. The second advantage is winter varieties. Now you can plant the winter varieties in the spring season only, but then you give that artificial chill treatment so that they grow fast and they will give you the yield. So with this, children, we completed all the concepts of the plant growth and development from NCERT. I think we covered line to line of NCERT. It will be helpful for you at the time of revision. If you want any other concept, text me in the comment box. So don't forget to like, share and subscribe my channel with your friends. Thank you.